3, 2.
Can I start? Okay.
Yes, please.
So can I start? Yes?
Yes.
Okay, so yeah, so welcome everybody. So it's a pleasure to have you all here. So my name is Angelo Rosa. I'm one of the co-organizers of this digital event. Welcome everyone. So just a few words about this event and the organization. So I'm part of the the CISSA people who organize this event, and I'm also the coordinator of the local SECAM node here at CISSA. This event is organized in collaboration with ICTP, and tomorrow Ali Hassanali will tell you about a few words about ICTP. So just a few words about SECAM, for people who don't know. So SECAM is a big European consortium who is putting together many institutions interested in computation, molecular simulations, and is probably one of the biggest consortium in the world. Normally, as a node, we organize such an event each year as a school for mostly for undergraduate people. We had the plan to do this year too in collaboration with ICTP. Unfortunately, for this COVID crisis, was not possible. So we decided to change the event a little bit and organize it as a group of talks online. So it's the first time for me actually to do these things. So please be patient if everything will not be perfect. So again, welcome everyone. We have a very nice list of very, very good people and very good talks. So for this first day, we will give the talk, the organizers. The idea is that basically to do seminars which are a bit introductory for the most topical talks of the next days. So in such a way that people who are not familiar with many of the topics, because you've seen that the program is quite broad, would get used to the language and then can follow easily the more specific talks of the next days. I have two advertisements to do, so please switch off the videos in order to save bandwidth. Okay, so that's good in order to avoid the technical problem. And also a more technical announcement. So the talks are recorded. So if there is someone that does not want to be recorded, just switch off the video and keep the video switched off during session. So you are allowed to ask questions by raising the hand, and then I will pick you and tell you that you can ask questions. So I don't know, for time being is all by my side. I don't know if Ali wants to add something or you.
Yeah, I know. Maybe I'll also take this opportunity just to welcome everyone on behalf of ICTP. So I guess all of you know. So ICTP is the Abdus Salam International Center for Theoretical Physics. We would have loved to have you all here in Trieste, but unfortunately, this is not going to happen. But you know, we're trying to make do with this virtual medium. We have ICP has a whole bunch of programs and a specific mission to develop capacity, scientific capacity in developing world. I'm not going to go into details here in the beginning of this event, but for those of you who are interested, definitely get in touch with me, and I'll be happy to talk to you. So I think we're already running late, so I'll just hand over to Angelo as the host.
Okay. Thanks, Ali. And so I think we can just move to Giovanni and to his talk.
Okay, so we share screen, right?
Yes, go on, please.
Okay, so can you see my screen?
Yes.
Shared? Okay, good. So good morning to everyone. So my name is Giovanni Bussi. So welcome to everyone. I'm also one of the organizers. And yes, as Angelo said, This is an experiment that we are doing. And it's also the first time that I co-organize a virtual event like this one. And actually, it's also the first time that I give a talk to a virtual event big like this one. So I hope everything will work smoothly. So the idea of my talk is to give you a flavor of what you can do to reconstruct biomolecular dynamics using molecular modeling tools, so molecular dynamics and experimental data. And in particular, I will focus on RNA dynamics because that's what my group is devoted to. And so at the very beginning of my talk, I will try to convince you to tell you why we decided to work on RNA, so why RNA is cool. So if you look at the central dogma of molecular biology, what you learn is that your genome is made of DNA, which stores all the information, and then it's copied, it's transcribed into RNA. And then RNA is used, the instructions written on RNA are used to build proteins, which are the real actors in the cell doing all the stuff. But actually, if you look at real numbers, this is only the fate of a very, very small fraction of the genome of complex organisms. And for mammals in particular, this is around 2% of the genome is coding for proteins, but the majority of the genome is coding for RNAs that have then other roles, other functions in the cell. And typically they have regulatory roles. So there are a lot of RNA molecules whose role is not just to store temporarily the genetic information, but they can do much more. And so it's very important to study them. So let me also give you a very short primer on RNA structure, just to set up the language. So the RNA structure can be analyzed at 3 different levels. So you have the primary structure or 1D structure. People call it 1D because you can represent it in 1 dimension as a string of characters. It tells you the sequence of nucleotides composing RNA polymer. Then you have secondary structure, which is the list of helices and loops, how they are arranged, which are the nucleotides that are Watson-Crick paired and so on. And this is also called the 2-dimensional structure because it's easy to put it on a piece of paper. But then, we are not living in flatland, and in reality, molecules can be arranged in complex 3-dimensional structures, and that's what the tertiary structure of RNA is. Okay, then that's the structure, but what about the function? Clearly, the function is related on the structure, but depending on the specific RNA molecule, it could be related to the primary, to the secondary, or to the tertiary structure. For instance, if you look at coding RNAs, those whose goal is just to store temporary genetic information, clearly the function is primarily linked to the 1D structure, so to the message stored on the RNA molecule. But if you look at non-coding RNA, the function could depend on the sequence alone, and as it is in some cases, or on the secondary structure, so which basically tells which nucleotides are paired and which are not, so which of them are available for further pairing, or on the tertiary structure, which is the 3-dimensional arrangement of the molecule. As I think it's easy to guess, so the 3-dimensional shape is particularly important when the function is exerted by interacting with partners, because typically when 2 molecules interact, they need some kind of complementarity in shape. And in this case, in particular, also dynamics is fundamental because typically molecules, when they interact each other, they have to rearrange a bit. So clearly for RNAs that are involved in functions like binding to proteins, to ligands, or doing catalysis and so on, the 3-dimensional structure and the 3-dimensional dynamics is fundamental. And that's where molecular dynamics comes in. And so molecular dynamics is a really powerful tool that can be used to model basically the system at the system of interest at atomistic scale. So every atom is represented as a point in space. Then you have some function that tells you which is the energy of the system, how it depends on the position of all the atoms. And typically this energy partly comes from chemical bonds, from angles, from torsional potentials, and so on. You have this is a very mature field where people have tried a lot of different forms. So to give you an idea of the kind of applications that we do are typically done using these empirical force fields, where you have interactions that are chemically motivated. You describe the system at atomistic detail, so every atom is a point. And we typically include explicitly water and ions, even if in many movies I don't show them for clarity. And unfortunately, these models do not take into account any polarization effect. So that means that the charge of each atom is decided a priori, the partial charge of each atom, and it has no way to adjust based on the context. And this is an important limitation. And if you want, the extreme case of polarization is charge transfer or chemical reactivity, and that's also out of question with this kind of approaches. Okay, so the good thing, that's the bad thing. The good thing is that these models are pretty fast and you can easily run several nanoseconds per day. I would say it depends on the machine that you have and how big is the molecule that you are studying. And as a tool, what we use is GROMACS, which is a very popular and free molecular dynamics engine, and Plumed, which is a tool to do enhanced sampling simulations that I will that I'm also co-developing, and I will advertise a bit better later. So why do I like molecular dynamics? So I like it because it gives you access to very high resolution, to your system with very high resolution, and it's virtually unlimited. I mean, it's we can access basically to as short time scale as we like, and also to very short length scale, so to the atomic scale. And in addition, we have access to dynamics, and it's relatively cheap in the sense that typically simulations can take a few days or a few weeks, depending on what you have to do. There are also drawbacks, and in particular, the timescales that you can access are not as long as you would like in many cases, and I will give you more details later on this. And then all the accuracy of your results is related to the fact that the model that you use is reliable. So I discussed already some of the limitations of these so-called force fields, and clearly, the better the force field, the more faithful will be the result. And then you have to remember that it's it's just a model. So it's a way to model your system, to learn a lot of stuff, but the most important thing is to compare your results with the reality. And in this sense, I like to quote this sentence by Vijay Pandey, that is, in science, as in life, it is very dangerous to fall in love with beautiful models. So we always have to validate our MD simulations against some experiment to have an idea if they really make sense or not. Okay, so the first problem is timescales. So which are the timescales that are relevant for RNA dynamics? So you will find around many pictures like this one in several reviews or papers. This is a picture which is customized for RNA dynamics, and where you can see that if you look at very simple dynamics like rotation around bonds, that's easily accessible on the nanosecond timescale. If you want to look at base pair formation and opening, that's more challenging because it reaches the microsecond timescale. And there are other processes like binding of divalent cations or even switching between different 2D structures that are totally out of reach for atomistic molecular dynamics, as you can see in the bar above. And that's why, in order to study this kind of process, you cannot just use plain molecular dynamics, but you have to complement it with enhanced sampling methods. And as I told you, for implementing enhanced sampling methods, what we use is a tool called Plumed. Okay, then there was also an issue that I mentioned with force fields. So what's the state of the art for, so what's the typical level? How accurate are force fields that are used to describe RNA molecules? This is just a bunch of pictures from the literature. I will not go into the details, but you have the reference if you want. These are just studies that, so from the group of Irgi Sponer and Tom Cheatham on tetranucleotides, and also from our group on tetradoops. I will just summarize this studies saying that the community is not very much satisfied with the current state of the art of RNA force fields. And there are many things that can be improved and should be improved. There have been recent improvements. I would mention a couple of relevant works, one from the David Shaw group and another from the Hirji Sponer group published last year, which are improvements, but still I would say that it's not easy to start a project using molecular dynamics on RNA and be confident that you will be able to reproduce experiments. And so in these years, as soon as I realized this, I slowly switched to, instead of just comparing with experiments, say using experiments as restraints in my simulations, which is a nice way also to understand more about how the experiments are done. So which type of experiments will I discuss now for RNA? Particularly interesting are experiments that allow you to access dynamics, so that take into account the fact that molecules are not like frozen in a single structure. And in particular, I would like to mention NMR. I'm not an expert of NMR, but to summarize very quickly, what you can access with the nuclear magnetic resonance is like distances between atoms labeled in some way, or torsional angles, or other geometric features. There is really a zoo of different techniques and people that are very specialized in each of them. The way to connect the formulas, the equations to connect the three-dimensional structure of a molecule to an experimental signal are kind of established. So it's kind of easy to compare with your simulation with NMR, or even to use NMR data as restraints. The only tricky part is related to the fact that NMR experiments are done in solution. This is technically a problem, but it's actually a very great feature because it allows you to access dynamics indeed. But the issue that comes in is that what you are measuring is not the property of a single copy of your molecule, but you are measuring some average over an ensemble of copies of your molecule. And so you have to keep that into account whenever you want to use NMR experiments as restraints for molecular dynamic simulations. Okay, so after this pretty long introduction, this is my optimistic agenda for today. I'm really happy to be interrupted. I'm not sure I will be able to see if people interrupt me, but I guess Angelo will be able to raise the hands and stop me if necessary. And I will be happy not to arrive to the end if I receive too many questions. So just stop me if there's something that it's not clear. So the first part of my talk, I want to show you how we combine experimental data, and in particular NMR, with molecular dynamic simulations. And I will give you a bit of the theory first. And then I will show you how we use this to study a very important non-coding RNA system. And then in the second part, if I can arrive there, I will show you some different way of combining molecular dynamics and experiments. And here I would say molecular dynamics is done in a semi-quantitative way, just to give hints about which experiments to do. And then the real work is actually doing the experiment. And this is something that we published recently. And it's a nice work where we managed to understand a lot of things about RNA and DNA unzipping dynamics. So let's see if I can arrive to that part. Okay, so the first issue is with the average restraints. So this is something on which the community of molecular dynamics simulation has worked a lot in the past 10 or 15 years. So the issue here is that so you have a molecule that can explore a number of different configurations. And whenever you observe something that is an average over these configurations, what you will see in your experiment, in your signal, is like an average over all these conformations weighted with their probability to be observed, P. So that's what you are expected to compare with. And now the problem is that this is a highly underdetermined problem. What do I mean? So if you have one observable, so if you measure the average distance between two atoms, there are a huge number of different possible probability distributions for that distance that are all of them compatible with the same experimental value. So in a trivial way, you can say if the average distance between two atoms is 5, it could be that the distance is always 5 angstrom, or it's half of the time 4 and half of the time 6. So how can you choose the probability distribution that is more reasonable among the list of all of those that are compatible with experiment? So there is a kind of consensus in the community that the cleanest way to do this is to pick among all these distributions the one with the largest possible entropy, and actually with the, to be more precise, with the largest possible relative entropy relative to a priorly decided distribution, which would be the Boltzmann distribution associated with the force field that you are using for your simulation. So in other words, what you say is that your posterior probability is among all of those that agree with the experiment, that would give you an average equal to the one proposed by the experimentalists, is the one that is as close as possible to the distribution that you would obtain just running a straightforward MD simulation with your force field. Okay, so how can you do this formally? So if you maximize the entropy and you compute its derivative with respect to the probability, and you can do some math, and it's easy to show actually that the posterior should be proportional to the prior multiplied by an exponential factor, which is e to the minus lambda times, and this f is the quantity that you are measuring. So let's say you are measuring the average distance between two atoms. This would be e to the minus lambda times the distance between those two atoms. That's the same as saying that the potential energy should be augmented by a factor, by a term that is linearly, linear in the quantity that you're observing with a prefactor lambda, and you have to choose this prefactor, and you have to choose this prefactor in such a way that the average over the posterior is equal to the experimental average. So this can be done iteratively. You can start with some value of lambda and then compute the average, compare with experiment, and then adjust it a bit until you have agreement between simulation and experiment. So some more comment on this method, this maximum entropy method. So here in this plot, I have like a histogram of some observed quantity. And let's say that my force field gives me the red distribution with two peaks, one here and one here with two different weights. And the average from the simulation will be some value here. And then I go to the experimentalist and it tells me, no, look, the average is here. So if you just add to your system a harmonic spring, keeping the distance equal to that value, you will have just a single peak in the posterior distribution here. If you use max ent instead, what you will do, you will rely on the peaks produced by the force field, by the molecular dynamics simulation, and just modulate them, so increase the weight of one and decrease the weight of the other until the average becomes compatible with experiment. So from this simple example, you can see that the amount of information that you retain from the original simulation is much, much bigger with respect to just enforcing a restraint in a simplest possible way. So you need a very good starting point, so very good prior distribution. And then you need also a lot of data. The more data you have, typically, the more reliable will be your result. So what I'm discussing here is maxent, but there are many closely related approaches in the literature. The group of Michele Vendruscolo has used since many years an approach based on replicas that is has been shown to be equivalent to this. And there is a method from the group of Parinello that was born as an enhanced sampling method, but can be casted also in a maximum entropy framework, and experiment-directed simulation from Greg Voth. And there are also Bayesian methods with very similar formulas and very similar properties. Okay, good. So if you look at these maximum entropy based methods, you have basically two manners to use them. One is to enforce, so to use your restraints during the simulation. And here you have a problem. You might have got that you have to choose these lambda factors to run your simulation, but you don't know the value a priori. So you have to adjust these lambdas during the simulation, and this is an This is done typically with an iterative procedure that is not straightforward, but this allows you to sample the correct conformations during your MD simulation. Or you can do it a posteriori with reweighting schemes, where what you do is you take your trajectory done with a possibly incorrect force field and then reassign weights to each of the snapshots so that you weight more the snapshots that are more correct, let's say, but using this maximum entropy framework in such a way that the weighted average on your ensemble will agree with the experiment. And okay, we also have done together with Max Bonomi and Michele Vendruscolo technical work where we compare the efficiency of these two possible approaches. So to make it short, so clearly reweighting is really nice because you do it a posteriori. So you take your 10 years old simulation or you take your fresh simulation, but you're weighted with the data sets from 10 different experimental groups. And every time it's very cheap because you just have to reanalyze your simulation. Whereas if you want to enforce your restraints, you need to have the experiments before you run the simulation. And clearly, if the experimental data change, you have to run your simulation again. Okay, so I don't know how much time I have yet. So I will try, we'll continue with this.
9 minutes.
Okay, so I think I will only show the first part. I try to go slow to be kind of didactical on this part, so I will show you this application now. So this is an application of this maximum entropy based method on a non-coding RNA. This is a signed B2 element that was identified in the group of Stefano Gustincic at CISSA some year ago. So this is a antisense RNA that is used to is used by the cells to enhance gene expression. So it basically finds a complementary messenger RNA, binds to it, and then enhance its capability to produce proteins by a significant factor. And the nice thing is that the experimental group working on this was able to identify, so they managed to identify first this element here, which is 100 and something or almost 200 nucleotides with this function. But now they are coming out with the idea that this hairpin alone is able to trigger this function. And so we decided to try to study the dynamics of this hairpin to have an idea of which are the structures that this hairpin could have. So what we did, or actually what the experimentalists did, they did NMR experiments first, NOEs, which means that you measure pairwise distances between protons, and you know which pairs of protons are close, which pairs of protons are far, or on average, let's say. And then what experimentalists typically do, they try to build a single structure that satisfies all these restraints, all these constraints. So if two protons should be close to each other, yes, that should be close, full stop. And then if you try to enforce all these signals at the same time, you can guess there could be some trouble because these signals could come from a mixture of different structures. So now if you look at the PDB that was solved using this experimental data, it contains 10 structures, it's shown here, but basically can be grouped in the like structures equivalent to this one and structures equivalent to this one with a small tilt in this nucleobase. Okay, so the first thing that we tried to do, we took these structures, which are experimentally refined, and try to run MD. And the first thing that we see is that, so the first structure agrees with all the experiment. We run MD for a while and suddenly we don't have agreement with experiment anymore. So the first thing that you might think is, okay, that's because your MD simulation is total crap. So you have a wrong force field. The system starts from the correct configuration, but goes into an incorrect one. That's actually not the case. If you look at the structures, it's not too difficult to realize this. And in particular, so here in this plot, what I'm showing is the distance that is measured between specific pairs of proton. And this, the chi, the glycosidic bond angle, is the rotation around the bond that connects the nucleobase with the sugar, which has some typical distribution that should be around here. Then if we take our MD simulations and then we use reweighting in order to assign a high weight to those frames that agree with the experiment, let's say, and the experiment tells me that the distance should be smaller than this horizontal line, then I will add a lot of weight to points which are here. But these points are in the tail of the distribution. If you look at the structure visually, you will see immediately that these structures are incorrect. They are really stressed. So in order to have this distance to be in agreement with the experiment, you make your nucleobase very, very stretched. You put it on the tail of the Gaussian, and this already suggests you that something should happen. Actually, what you can expect is that in reality, there is a mixture of this structure and of another structure that is completely different. But once averaged correctly with this one, would give you the experimental observation. Okay, so in order to see these rotational torsional angles, you need the enhanced sampling. So here we used some variant of metadynamics and replica exchange that if you want, you can find in the paper, the details. During the simulation, we also enforce this, the agreement with these experiments. We see dynamics. And then when we analyze with the same metric as before, our simulation, we see that from time to time, so some of the structures goes to a different rotamer where this torsional angle is flipped to a scene conformation, which is expected to be rare, but not impossible. And indeed, that's able to explain the experiment without the need to resort to very uncomfortable position for the nucleobase. Okay, so how can we then rationalize our results? So at the end of a Maxent procedure, what you have is an ensemble. You have a lot of structures, but you would like them to be not too many, not thousands, because you don't want to watch 1,000 structures. So the first thing we did is a clustering analysis. And also Sabine Reiser, who was the postdoc working on this project, worked a lot in doing these cool pictures. This tells you basically how heterogeneous is the ensemble that you expect to exist and to be in agreement with the experiment. Okay, there is one important comment about this clustering, because we struggled a bit on doing clustering correctly here. What we wanted are clusters that are really homogeneous structurally. So with basically the same NMR signals and structure which are similar to each other. And standard clustering methods do not have this feature. And so what we did, we decided to use a homemade, very expensive method based on maximum clicks that work pretty well. But there is a very nice paper published this year by González Alemán et al., where they actually show that quality threshold clustering, which is a method published in the 90s, can be used very effectively to obtain exactly this. So clusters that are structurally homogeneous. And that's not true with some of the methods that are used very much in the literature, as it's explained in this really nice paper cited here. Okay, so still, okay, we have clusters, but there are too many. I really don't want to watch all of them. So Sabine Reiser had the idea of trying to combine maximum entropy with maximum parsimony. So let's try to, instead of using all the structures that we have, let's try to only use structures coming from one cluster. Can we reproduce the experiment? The answer is not. So let's try to use structures from 2 clusters, 3 clusters, and so on. And what she discovered is that for this specific system, what you need is 4 clusters, which due to the way we define them, they are different from each other. So they contain structures with different annotations and different base pairs and stacking patterns, with this specific population. So the answer is not unique. You have different possible sets of 4 structures, but the nice thing is that you can really find out which are the structural features that are required to reproduce experiments. So you can see that you need some of this guanine to be in a scene conformation, and you need some of these AU base pairs to be broken. And that's very interesting, because for this we have kind of independent validation. We have other experiments that we did not use in this calculation that suggests that this pairing AU here are at least transiently not formed. So I would say this is a kind of independent validation of our result.
Then you should conclude in the minute or so.
Yeah, okay. So just summarizing this, okay, I will not summarize. Let me just make a small advertisement. So if you are interested in learning more about enhanced sampling methods, we are organizing Zoom masterclasses. And I think Max Bonhomme will tell you more about this when at his talk. There will be Zoom lectures with a limited number of participants. The participants will interact through a Slack workspace, and the deadline for application is soon, is in a couple of months. And okay, now let me jump to the very end of my talk, just to show you the pictures of the people that did the job. So this is an old group picture, but I needed this because this is a work, the work that I showed was made by a postdoc that now left my group with Sabine Reiser, who is here. And then I want to thank also the experimental collaborators for the work that I show you, that are Stefano Gustincic and Silvia Zucchelli, who disappeared last year. And then thank to all of you for your attention.
Thank you very much, Giovanni. I send also my reaction. And so yes, so questions. So as I said, if you want to make a question, so just raise your hand and then I will switch off your audio. So there is a thank you very much from Davide Bassani.
Don't be shy.
Yeah, don't be shy. Thanks a lot. Can't find the raise your hand. May I speak? Sure.
Yes, this is Shravani from India.
Hi.
Hi, nice to be in ICTP after a long time. So I have a question, very nice talk. I just needed to understand, I do see in the literature, not only maximum entropy, but also maximum caliber models. So what is the difference between the two?
Okay, so maximum entropy is for ensemble averages. Maximum caliber is for dynamical trajectories. So in a nutshell, it's the same, but instead of being applied to averaging between conformations, it's applied to averaging between trajectories. So again,
so if I have a non-ergodic system, then only there will be a difference in the two predictions. For ergodic systems, it should not matter, right? If I have long enough sampling.
I'm not sure this is a correct interpretation. So the point is that with the maximum entropy, what you obtain is ensemble averages, but you don't obtain time correlations. Now, let's say that you have an experiment that measures the time correlation function of an observable. Then you can use the maximum caliber in order to enforce that time correlation function in your simulation. So it's like the difference between enforcing an ensemble average and enforcing some time-dependent ensemble average, as it would be a time correlation function. So it's really, I would say, it's related to the kind of experiments that you use, not on the effect on the ergodicity of the system.
Okay, okay. And I have a second question. If you know the rate constant of a given, associated with some kind of a conformational fluctuation in the kind of systems that you're talking about, so which enforcement is going to be more appropriate?
If you know rate constants, I would say you cannot use maximum entropy, you have to use maximum caliber, or in other words, maximum caliber is just, is nothing less than, from my understanding, nothing less than maximum entropy applied to trajectories. So, but you have to use maximum caliber to enforce rates, as far as I know.
Thank you very much.
Giovanni, there is a question by Sharon RT. Is it said is or she is asking, is it possible to use experimental restraints to then refine the force field?
Yes, it is possible. So this maximum entropy framework is not designed to do that, because the correction to the force field is going to be by construction, the one that changes the least, the distribution. So as I said, if you are measuring a distance, you will correct directly that distance. Whereas when you want to refine a force field, typically you choose a priori based on some other chemical intuition, the functional form. Let's say you want a force field made with torsional angles. And then you can refine those torsional angles in order to get agreement with experiment. But that's not within the maximum entropy framework, because you will not maximize entropy anymore. You will maximize agreement with experiment directly. I don't know if I answered to your question.
Yeah. Other questions? I know she, yes, no. Are there more questions? No question. I cannot see any. Yeah, don't be shy, ask questions. Well, okay, I don't know. If there are none, I think we can thank Giovanni again for the talk.
Thanks to you, and so I will stop sharing now.
Then I think we can move to Edgar.
Yes, can you hear me? Hello?
Yeah, we can hear you, Edgar. Yes.
Okay, very nice. 1 second, let me try to share screen. Right, you see my screen?
Yes, we do.
1 second more, you see my pointer? Do you see my pointer?
No. Ah, yes, yes.
The red thing, okay.
Dot,
yeah,
yes.
The red dot, exactly. Okay.
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 Please, go on.
Okay, so welcome everybody. I'm one of the co-organizers of this online activity. I want to thank the other co-organizers, especially to Angelo, who took care of a lot of work for this to happen. As Ali said before, I also work in ICTP, so it's a pity we cannot meet in person. But okay, we can do what we can do at this moment. And I would like to invite everyone who has questions to make questions during my talk, especially young people, because I'm going to discuss about stochastic thermodynamics, which is not the main topic of this event. So there will be a lot of new concepts for many of you. And in particular, I will discuss on something we call Martingale, which is a tool used in finance that we are using in the recent years to explain fluctuations of thermodynamic systems like the ones. Well,
sorry to interrupt. There is a small window in the middle of your slide.
A small what?
There is like a small gray window that that is overlapping on your slide. I don't know. It's written no build effects.
I don't know. Do you see it now?
Or maybe just move down. Okay.
Okay. Well, let me start with a review of thermodynamics. This is what it's in many textbooks in classical thermodynamics. So the first law says that you can get something for nothing. So energy, as is illustrated with this stone, is transformed into two different forms of energy. One is heat that is dissipated in the surroundings, and the other is called work that is used to move things like this bucket we have here. The second law, on the other side, it discusses the irreversibility of thermodynamic processes, and it says in few words that you can't even break even, meaning that, as I show here in this with this glass, you can processes tend to increase the disorder of the system or of their environment following this law. So you can, in general, the entropy of a system increases, meaning it becomes more disordered, and also the entropy of its environment. So you can make a system more organized by reducing delta S system. So the system entropy reduces, but there is a price that is you have to pay this price in heat dissipated to the environment, which increases the entropy of the surroundings. Right. So in the recent years, there has been a lot of progress in doing thermodynamics of smaller systems, like, for example, a DNA hairpin, a colloidal particle, or a nanoelectronic system. Here I show an illustration of a colloidal particle that is driven out of equilibrium because there is an external force pushing the particle, but it's a small particle, so it prevents fluctuations.
well, I'm also a co-organizer of this activity, and I want to also thank Angelo because he made a lot of work for making this work, and thank you all for being here. I'm going to talk about a topic that is somehow transversal to all the
Alex
talks that we are going to see.
Alex,
he can't.
How clustering technique are applied to molecular data.
Alex.
First of all, I want to say what is clustering. Clustering is supervised machine learning.
Maybe we have to change the speaker
By
at this
supervised
moment.
machine learning, we understand something that deduces the
Can someone mute Alex?
Without using any a priori classification.
non lo so.
Okay, all fine. Can you hear me?
Yes. So sorry, there was probably misunderstanding.
All right, let me go on. So I was talking about this particle moving right and left with fluctuations, which is the typical example of stochastic thermodynamics. So we do thermodynamics with few degrees of freedom. For example, we aim to understand what is the heat dissipated by the particle when tracing a particular trajectory. This type of formalism was introduced in the 98 by Ken Sekimoto, who, by the way, will be giving a talk in this activity. And he introduced a formalism to describe what is the heat along a single trajectory, or what is the work along a single trajectory of the particle. So these are new concepts developed in the last 2 decades. And there's also the concept of stochastic entropy production, to which I will discuss in detail in this talk. And there's a second law, which is now for ensembles of trajectories. So you can have a trajectory of the particle going against the force, and this is interpreted as a negative stochastic entropy. But on average, the particles, if you look at many trajectories, they will move more towards the right than towards the left because there is a bias. So I will go into this in more detail later. This is a typical example of a model that we study in stochastic thermodynamics. So we have a chemical reaction, with reactants and products. We have different chemical potentials, and thermodynamics tells you that you will have a flux in the direction of the gradient of the chemical potential. So you will have, for example, this reaction happening in this direction. In stochastic thermodynamics, we say everything can happen. So we assume that all reactions and the time reversals can happen with different rates. And a key assumption in our field is that the rate of a process and of the time reversed is related by what is called the environment entropy produced along one of these transitions. So this is for one transition, but as I was telling you, you can look at a particle that is drawing a long trajectory so that you will have many transitions. So when you collect these many transitions, you can build probabilities of trajectories given an initial state and probabilities of the time reversal trajectory given a final state. If you take the assumption of the equation I gave you before, these two will be related by the environment entropy change along this trajectory. Similarly, you can do the same with the system entropy. This is not the Shannon entropy, but it's the stochastic version of the Shannon entropy. Just the logarithm of the probability to be in a site xt at time t.
Edgar,
Yes.
No, there is still this technical issue with your slide. There is, on the screen, it appears like a small gray window on top of your presentation. On the top right corner. Yeah, maybe try to.
Okay. I'm closing everything I have open in my computer. So
yeah, because otherwise it goes on top of the formulas. Then we don't see things.
Yeah, I don't see anything. That's why. Okay. Is it fine now?
No, it's still there. I mean, I don't know. It's written no build effects.
I don't know.
Is group?
All right.
It looks like some. What are you using?
I'm using Keynote as always, and then.
It looks like some
And
window.
then I'm just
Maybe
okay.
Edgar, right now there's something on the right called build order.
Yeah.
Can you close that?
Okay.
Close that.
Okay, sorry.
That's the problem. Yeah.
Okay, give me 1 second. This is the animation panel in. All right. I guess now it's fine, no? Is it fine?
Perfect.
Yes.
Yes.
Okay, thank you. Sorry. All right. So this is the definition of the fluctuating system entropy. And if you sum this entropy plus this entropy, you get what we call stochastic entropy production, which becomes the Boltzmann constant times the logarithm of a full trajectory divided by the probability to see the time reversal trajectory. So this is a measure of time irreversibility of a process. And we often write it like this. So the total entropy associated to a given trajectory is the logarithm of the probability to see the trajectory divided by the time reverse probability. So the probability to see the time reversal trajectory, right? This is a formula that we use in stochastic thermodynamics to study the irreversibility of different processes. Here are examples of systems that are paradigmatic. For example, we have a periodic potential with an external force. We say this is a non-equilibrium, but it's steady state. So this is an external force that is time independent. So we are not changing the Hamiltonian in time, but there is an external force pushing the particle such that it has a net current. But there are more complicated models, for example, Feynman ratchets with 2 thermal baths, or active matter, which is attracting a lot of attention in the last years. So stochastic entropy production has some universal properties. One is that if your system is in equilibrium, it doesn't fluctuate. It is always equal to 0. So its average is equal to 0. But the most interesting part comes out of equilibrium. So out of equilibrium, this is a fluctuating quantity, and it can even be negative. You can have an experiment where the particle is climbing a potential, and this we interpret as a negative stochastic entropy production. But on average, when you average over many experiments, you will have always a growing curve. This has been seen also in experiments, and you can see negative entropy in electronic experiment that I will discuss later. Right. So in the last years, there has been a lot of research on finding universal results for entropy production, work, heat, etc. So first of all, let me say that this looks like a purely statistical object. So in principle, it is, because you just have to measure probabilities of trajectories. You don't need to know anything about the physics below. But if you take a physical system, like an electrical circuit, you can measure the growth, net growth of this stochastic entropy, and you see it's related to the heat dissipated in the resistor in this experiment, for example. And as fluctuation theorems, as I said, these are theorems, these are relations that are universal. They are common for many physical systems. And one is very important one is the detailed fluctuation relation shown here that says that at a given time t, it is more likely to see positive entropy than negative entropy by this relation, which implies a so-called Jarzynski equality. All these are fixed time properties looking at your system at a given time. But I was interested in the last years in finding other type of new properties for which there was not much known. For example, the random time. So what is the first passage time of entropy production? How long it takes to produce 1 kBt? Or what is the extreme value of entropy production? We say it's negative. It can be negative. So how negative can it be? And these have obviously important consequences for example, enzymatic reactions, which we know that happen at fluctuating times, or in polymerization processes where you can have backtracking events, and this extreme entropy could be related to the extreme excursion of a motor. I will discuss about this in a while, right? So this is also to motivate, I think. And maybe now go to the key concept in this talk, which are martingales. These are processes used in probability theory, also in gambling and finance. And they have interesting mathematical properties because they provide a shortcut to tackle extreme values and stopping times. So we can have tricks to calculate extreme values in an easy way somehow. And they have been surprisingly unexplored in stochastic thermodynamics until this paper and also our work. It started a new research line on thermodynamics with Martingales. So it's very surprising because they appear in all the books in probability theory, gambling and finance, but there was no link not such a strong link in physics until recently. What is a martingale? So a martingale is a process that we say a martingale with respect to xt is a real valued function. It's a real process that is bounded. And the most important thing is this condition here. So if you know x from time 0 to time s, this is a martingale with respect to x. What you expect in the future is the last observed value of the process. So it's a process that has no drift. For example, Brownian motion is a martingale, and fair financial markets are martingales or fair games. To give you an idea, this is an illustration. So you look at the process and you know the history up to time tau. If the process is a martingale, what you expect any time in the future is that its average value is exactly at the last observed value of the process. This is what this is meaning. So it's a process without drift. All right. And this has been applied very much in finance. So let me ask the following question. Can a gambler make fortune in a fair game by quitting at an intelligently chosen moment? So you have a process and you say, okay, for example, this will be the money I have in the stock market. And I will say, okay, I want to win. So all I need to do is wait until this process reaches a good benefit. I don't know, 10,000 euros, for example. No, you just wait. So you would make profit out of this action. However, the problem is we have finite money. So at some point you will enter, if you have Brownian motion can go infinitely low. So you can get infinite losses. So you would make profit, but this is not possible in gambling or in finance. However, you would say, okay, I want to win. I want to stop if I get 10 euros as a revenue, or if I'm losing 5 euros. So I do a two-boundary first passage problem, and I wait. It turns out that if you have a martingale, by doing this, there is no combination of thresholds by which you will win. So at the end, at the stopping time, the average of the process is the same as at the initial time. So on average, the gambler makes no profit. So you cannot make profit with martingales. This is the main message. So a gambler cannot make fortune if it cannot foresee the future and it has access to a finite budget. So this means it's using a stopping time. It cannot see the future. And second, it has a finite budget, is uniformly integral. So this is a bit technical, but mainly there was this theorem by Doob that says if you have a martingale, what you expect at the stopping time is the initial value. Okay, this is called Doob's optional stopping theorem, and it's valid for any stopping time, not only a two-boundary first passage problem. Okay, this is a very powerful result, true for all stopping times. So this is a big class of first passage times, let's say. And we found in 2017 that there is one property of entropy production that is martingale. First of all, we found that entropy production itself is a so-called sub-martingale. So you have this condition. If you know the past, you know that in the future, you will grow, given any previous knowledge. And this implies the second law, because if you put tau to 0, you know nothing, so it implies the second law. This came from finding that exponential of minus the entropy is a martingale, so it has no drift. So we have this condition that I'm trying to show you. This one is a martingale, and this, if you set tau to 0, this implies Jasinski equality. So it's very nice. This looks a bit mathematical, but this open up because you can take a book of martingales and look at the mathematical properties and apply them to physics. And this was what we did next. So we applied properties, mathematical properties of martingales to get into the stopping times and the extreme values of entropy production. So in the stopping times, we found, applying the martingale theory, very surprising results. One was that if you put two absorbing boundaries and you measure the time it takes to reach the top and the bottom boundary, you will have more hits in the top than in the bottom boundary. But the time it takes, so the distributions look the same. The histogram has more counts, but the shape is the same. So there is a symmetry in how long it takes to produce entropy production. There is this symmetry for the times, which was for us extremely surprising. So you are going in this direction more than in here. But given that you reach the boundary, you take the same time to reach the top boundary with a drift than to reach the negative boundary. So this was a very surprising symmetry that we could test with an experiment. So you have the particle here, you put a boundary here and a boundary here. And of course, it will fall down because you are pushing the particle more. But if you measure the time to reach the boundary in the bottom and in the top, they are the same. They have the same distribution. And this is a new result that we found using Martingales. Moreover, we applied this theorem that I told you that you don't gain with Martingales to entropy production. And we had now a fluctuation theorem at random times. So any gambling strategy that you use, you will have no gain for e to the minus s. You will have a gain for s because you apply Jensen inequality and you get, there is a second law at the stopping times. And this is for any gambling strategy. You can put two boundaries, and the boundaries, you can put them in any positions you want. It's a very, very, very general result. Right, and this is an illustration. So in a fluctuation theorem, you have systems in a non-equilibrium steady state, and you wait for some time. You let them evolve. You let them evolve dynamically up to a given time, and you collect the entropy at the final time, the entropy production, and if you sum them, it's positive. Now we say something else. We say we put a criterion. So we put, for example, a ring, and we track the systems until they cross this ring. This is at random times. Each system crosses at a different time, and we measure the entropy production at this crossing time, and there is a second law, even in this. The nice thing is, that this one is a particular case of this one, because one specific stopping time could be you have your clock and you wait until your clock ticks 3 seconds. So it's generalizing the previous results. Right. And now I think this is even more exciting, are extreme values. So we are looking at what is the negative record of entropy production. You look at entropy production to a given time, and you'd like to know what is the negative record. What was the minimum value in a finite time? With Martingales, we can calculate the statistics of, we call this infimum, because there is a theorem by Dub, by Jean-Phil, because he stole it from him, which tells you the distribution of the maximum of a supremum is bounded by its average at the final time. You can apply this to entropy production and get these very nice results. The first is a bound on the full distribution of the minimum. And the second is that the average minimum cannot be below minus Boltzmann constant. So there is a lower bound, fundamental lower bound for the minimum of entropy production on average. And this is very, very general. So these mathematical results, you can take a theorem and see what are the assumptions of the theorem. And these assumptions become conditions for the physical process. These are valid for Markov chains, for Langevin dynamics, for continuous time Markov process in a steady state. It's a very general result. It's so general that we went to a lab and tried to test this in an experiment. This was a condensed matter experiment. It's an electronic double dot. So we have two islands, and there are electrons. These are very low temperature. Electrons that can tunnel to these islands. The electrons see each other, and they have a Coulomb repulsion. So all in all, this behaves like a four-state system in which either you have no electrons, you have one electron on the right, you have one electron on the left, or you have two electrons populating the islands. In this system, you can measure entropy production, stochastic entropy production for different biases. When you bias very strongly the system, this grows very fast. And from here, from these trajectories, measure these extreme values. And it turns out, measuring the extreme values and building the distributions, that our theory is really good. It's really providing a bound to the extreme excursions, the distribution. And moreover, if you do the average minimum, at very long trajectories, it cannot be below this bound we found, the -1 bound for the infimum. You get closer when you are closer to equilibrium, 25 microvolts. When you drive strongly the system, you don't saturate to -1. And this is something applied later in biophysics. So now let me go a bit more on the topic of the conference, which is an application to biophysics. So we use this theory of extreme values to relate molecular motor fluctuations with random matrix theory. This is a surprising result derived by my student, Alexander Guillet. We were looking at a simple model of a molecular motor. So we just have a walker that walks to the right with jump rate k plus and backwards with jump rate k minus. And this is in periodic boundary conditions. So time is continuous, but space is discrete. We define here A is a cycle affinity, is the bias parameter, which is related to the force, net force applied to the motor, the length and kVt. And there is also a symmetric parameter, it's called rate, that appears as a prefactor in the two rates. This is a simple model. So in principle, one could be able to calculate everything analytically here. The nice thing is, if this is in a ring, all states have the same free energy, this one, this one, this one. We have a uniform distribution in the ring. So the entropy production becomes just the work. And this is FL times X, simple. And therefore, the entropy production in this model is proportional to the position of the motor at time t. Proportional. This is not general, but it is a simple example to illustrate our results. All right. So this means that when you jump with the drift, you dissipate heat, so you produce entropy. And when you jump backwards, you are reducing entropy. So it's like you have a particle rolling in a linear potential. And when you roll down, you dissipate heat. When you roll up, it's because you are taking energy from somewhere, from the above. The questions we wanted to address is you have the trajectory of the motor, single trajectory, and we look at what is the running minimum, what is the minimum up to time t, and the running maximum. We'd like to understand what are the statistics of the running maximum and of the running minimum, and also of the running maximum of the entropy production and of the running minimum of the entropy production. These are important in biology in general because to understand robustness and resilience in biological systems, which are subject to failure. Like, for example, there's here a microtubule that there's an extreme event in which the polymer it starts to enter in catastrophe, or sperm navigation is an extreme event as well. No? So we think that understanding resilience and robustness is important in biology, not only averages. So the first thing we could derive in this model is the long time extrema. What is the long time minimum of the position of the walker? And first of all, we find this is the distribution of the minimum is a geometric distribution. And second, the average minimum has this expression. So it only depends on the bias parameters. And when A is 0, this goes to minus infinity. So at the stall force, the extreme value diverges. The average extreme value, which is this line, diverges. However, this doesn't happen with entropy production. This entropy production is A times this. So what happens is the following, that this is bounded. It's not bounded by minus infinity, it's bounded by minus the Boltzmann constant, sorry, which is what we showed in the previous work, actually. So when you make the bias stronger and stronger, the long time minimum gets closer and closer to 0. When you're close to equilibrium, you are here. So more importantly is that not only we can see the average long time values, which are here, but that my student could derive a very nice expression for the full curve, the finite time average minimum, which is given by this formula. It looks a bit scary, but I'll show you a very nice way of expressing this formula. So the main point is we found there is a symmetry, min-max symmetry, between the minimum of entropy production, this excursion, and the maximum. So this is the value of time t minus the maximum, has the same statistics as the minimum. And this is related by this formula. Interestingly, we found that this formula can be rewritten as an integral over times. So this is like a relaxation spectrum, and where the relaxation spectrum is given by the so-called Marchenko-Pastur distribution. This is a distribution that has it has a finite spectrum. It has this asymmetric shape. This is it's called Marchenko-Pastur, and it appears, interestingly, in random matrix theory. So it turns out the following, that if you want to calculate the extreme value, you can do a very nice shortcut. You draw a random matrix. So this is a matrix which is size m times n, and its entries are random numbers. And they can come from Gaussian random numbers, for example, with a given rectangularity. So the size, the asymmetry in the size is related to the asymmetry in the bias of the Walker. So you plug in random numbers in this matrix, and you do construct the Wishart matrix. So you multiply the matrix by the transpose, and you divide by the number of columns. Then you diagonalize the matrix, you get the eigenvalues, and it turns out that if you make this sum, exponential of minus t divided by these eigenvalues, this gives you the average minimum.
Edgar,
the matrix, that's, yes.
You have 3 more minutes.
Okay, fine. I have more stuff, but I'll summarize soon. All right. So it turns out that you can write you can compute very efficiently extreme values of the molecular motor by drawing very small random matrices. And this is what we show here. The black line is the exact calculation, and the points, for example, is taking a 4x4 matrix, just one matrix, making the eigenvalues and making the sum. And this gives you this very nice approximation to the extreme value. So we got very excited about this result, and we are trying to fully understand it because it's a surprising connection to us. I don't have much more time. Just to tell you very briefly, these ideas can be applied to more complicated models where you have a spatial coordinate and a chemical coordinate. For example, ATP consumption and the position of the motor. And okay, I don't have much time, but recently, very recently, we introduced what is called gambling demons. They are different to the Maxwell demon. It's illustrated here. It's a demon that is gambling. So it can either stop the process at a fixed time or stop when it has gotten a very good revenue. So it means it is using, okay, this work is done by Gonzalo Manzano here at ICTP. You can stop, for example, the work at a given time only if the work didn't cross a given threshold. So this is similar to what I was telling before on the stopping times, but now with driving. Before it was steady state, now there is driving. And okay, I finish soon. It's just that we derive this second law. Work minus free energy at stopping time is not greater or equal than 0, but it's greater or equal than something else, which if you have a question, we can discuss in the discussion tomorrow, which is related to the asymmetry of the process. And we could find in an experiment that you can go below the free energy by doing these gambling techniques. And this we could see very well recently in an experiment. The more irreversible you are, the more you can gamble and more you can go far from the second law. And this is very interesting. You can see this on arXiv very recently. Okay, just to finish, Martingales in the air. So there are different authors that are following this approach. There are here good examples. Ken Sekimoto will give a talk about it in this activity. We are writing a review. I put here to be finished in 2050 because it's taking us a long time to finish. And I want to thank you for your attention and also to different collaborators on my current team for the great work. So thanks a lot.
Thank you very much, Edgar. So as before, we are open to questions. As before, don't be shy.
Yes, even if the question is conceptual, anything, please go ahead.
So maybe I just start to break the ice. And this last part, Edgar, sorry, I didn't understand what is this delta? I just missed it.
Yes, so this is okay, 1 second. The delta, it's okay, 1 second. delta is information theoretic quantity, which is the logarithm of the ratio of, this is the density at time t in the forward process, and this is the density in the backward process, because here it's not a steady state, so it's a driven process. So you drive the process from time 0 to time t, and you look at the snapshot at a given time. This one is the same snapshot in the backward process. So you are looking at the density of the process in the same time, but in the forward on the backward process. So this is the density. For example, there is a process that goes from time 0 to time 10, and this would be the density at time 2 in the forward process, and this is the density at time 8 in the backward process.
Okay. So it's a measure of the things diverge.
Yeah, so this is the forward process, and sorry, one question. You start from 0. Okay, you start from 0, and you finish in tau. So you're looking at a given time in the forward process. Sorry, my drawing.
Yeah, no.
And the forward process is flowing in this direction.
Okay.
And the backward process will flow like this, okay, in this direction. So you are looking here at this time in the backward process here.
I see. Okay.
Forward and backward. When the process is very reversible, the statistics of the process here in the forward process will be different than the statistics of the process in the backward at the same time instance. So this is measuring the density of X at time t. This one, sorry, this one is here. And this one, I'm just messing up the colors, but this one is here. So it is measuring the probability to be in a given X at a given instance in the forward and the backward process. When you are in equilibrium, they are the same. But when you are out of equilibrium, the driving processes generate asymmetries in the distributions. So that's why we are calling stochastic distinguishability. Yeah. The distinguishability between the forward and the backward process.
Thank you. So we have, I see that we have a hand raised by Marcelo Albuquerque. Please go on.
Okay, thank you. Thanks for the talk, Professor. Nice talk. My question is about the bias to the system. I don't know if you understood what you said previously, but you said that the system, I don't know if it would be biased or if it could be biased. I would like to understand.
Biased.
Yeah.
Yeah. So this could be, for example, a motor pumped by ATP, and there is a chemical reaction that is, and there is an asymmetry. For example, here, there is a potential here that is asymmetric, and there is a force, ATP, that is pushing the motor more in one direction than in the other. In the way we describe, this can be anything. So this is a net force on the motor. For example, there could be someone pulling from the motor. For example, you can have a motor in an optical tweezer, and you can pull from the motor in the optical tweezer. This means there is a bias. So the motor is walking more in one direction than in the other. It has transport.
Okay, thanks. So we will have to take into account the force that you are putting.
Yes.
System, right? Okay.
Yes.
Thank you very much.
All the models I discussed have a bias. They are non-equilibrium.
Okay, thanks. Thanks.
Well,
excuse me, I have a question. You mentioned about macrotubules in one slide. I don't remember. Could you please explain more? Because my work is more is about macrotubules, and so please explain more.
Course,
yeah, thank you.
Yeah. So here the model I take is a motor that moves forward and backwards stepwise. So this is a forward step. This is a backward step. And the motor is biased, so it makes more forward steps than backwards. And I'm interested in looking in this process, the black one, what is the maximum of the process, what is the running maximum, or what is the running minimum. So the blue process is what is the minimum up to this time. If there is a minimum now, if the process goes below, the minimum has changed. This is like the record. I know, records in athletics. So this was the shortest time, but now there's a new athlete that has a shorter time. So this goes down, right? So we are looking at records in stochastic process. This one is a stochastic thing. In microtool, this type of events, downtrends, are very important because you can build a trajectory with the length of the microtube. So now I'm not looking at where is the motor. I'm looking at where is the tip of the microtube. When it grows, this goes up. And when it shrinks or has a catastrophe, the length will decrease, no?
Yes.
Okay. So all in all, you have a process that is doing like this, fluctuations, like the black process here. So here, the extreme values are very important because negative extreme values are catastrophic events.
Yes.
So if the extrema goes very, very low, here is catastrophic because it means the process will, the microtubule will be dislodged fully. So I think there is a lot of room for application of our results that are general to these models. It can be applied as well.
Thank you very much.
You're welcome.
Other question? Yes, so, sorry, I have one more question myself. So regarding these details of this model, you said that this is discrete in space, but continuous in time. Why is it continuous? Is not just a random walk? I mean, on a
Yeah, this is a biased random walk in a lattice in continuous time. Yes.
But yeah, why it's continuous in time? It's not because you're doing some kind of.
Well, I assume time is continuous always, and I put discrete space because it's a coarse graining effect.
Yeah. Yes.
But so mainly what we are doing in this time series is a Gillespie algorithm. So we run at random time until the next step. But this is a good description, I think, for single molecule experiments when you can distinguish states in the model.
Yes.
Of course, let me just say one thing more. Of course, it will be, it is more precise to me to have something like this, like you have your motor here, and you can either do a displacement by a mechanical force, you can consume ATP without moving, which will be this, or you can consume ATP and move, which would be this event. So using discrete, to me, the best model is continuous space, continuous time. But using this type of approach with discrete is useful when in an experiment you can only see part of the dynamics. You can see only coarse-grained states of the system.
Okay.
But I would take that continuous time because I think it's more, it makes more sense. Yeah.
So we have another question from Katere Azizi. So please.
Thank you. Very nice talk. You showed some graphs on the entropy evolution in time, which was decreasing. So I was curious: is it for the isolated systems, or is there any enthalpy effect, or it's just so they're
here?
related and
yes, this is a good point. Here, the description that we do in stochastic thermodynamics is something more similar to what you see here in 1 second in this figure. This is the paradigmatic model. So we have a colloidal particle, is the gray particle. This is in a potential, is this black line. So this is the colloidal particle. It is in a potential. There is an external force, and there is a bath. I'm not putting the bath molecules here, but this is full of other particles, green particles that are around, that are interacting with this particle. So I'm not talking about an isolated system. Of course, all together will be an isolated system. But we look at the dynamics of only of this guy, which you can describe by this type of models. It's a Langevin dynamics where there is a bath, which is this noisy term. In other words, I think it was better this one. We are doing thermodynamics on this colloid, but this is the entropy that we look is up. The entropy exchange between this colloid and the environment, not of the full fit. Okay. It's a classical system. It is canonical. It is in a thermal bath. It is not an isolated system. Of course, to do this simulation, you need to isolate the box. But in stochastic thermodynamics, we just see this red particle. We don't see the others. We want to do thermodynamics on the trajectory of the red particle.
So we look at the entropy of individual particles, and then we will see some decreasing.
No. It decreases because this particle is moving backwards. So we live in the world of Langevin dynamics.
Yes.
We live in this world. For us, this is what is our world. So in this world, you can have events where the particle climbs up, and this is because it's absorbing heat from the environment. But we are not tracking the heat exchange with each individual particle, but we are living in this description, which is noise. The noise accounts for an average force of the bath molecules. So it's an effective description. It's a mesoscopic description, what we do. Yeah,
Thank you.
The entropy. Okay. Hope it's clear.
Thank you.
Welcome.
Okay, thank you very much. I think we stop here. Thank you, Edgar, for the very nice talk. And so I think now we move to Alessandro.
Hi.
Okay, Alessandro, you can just go on.
I can share the screen.
Yes.
And all right. Fine. So.
Okay, great.
Please tell me if you can see my presentation.
Yes, we do.
All right, great. So my name is Alessandro Laio. I'm also working in CISSA. I have been working for quite a while on the development of advanced sampling techniques, even if more recently I am more focused on data analysis and data science tools, also for analyzing biomolecular systems. But okay, today I will give you an overview of, well, not of advanced samplings in general, because this would require 5 days at least, but at least on the reason why advanced sampling is necessary. Okay, so, well, our dream as scientists, well, let's say my dream as scientist, everybody can have his own dream, but okay, one of my dreams as scientist is actually, has always been trying to understand from a physicist's point of view, how biomolecules work. So these biomolecules are amazingly interesting machines, which actually are extremely efficient. They do incredibly precise tasks based on what? Based on the laws of nature, of course. So they basically convert thermal fluctuations into ordered work in a manner which is absolutely amazing, and we would like to be able to copy in order to, well, for example, design nanomachines. So this thing is understanding really how biomolecules are working is actually a big challenge for nowadays science. And well, and molecular dynamics has been and is going to be one of the key tools for addressing this extremely important challenge. Okay, so what is molecular dynamics? We have already heard a lot about so in the talk by Giovanni. So given a potential energy surface, one solves Newton's equations of motion. So very, very easy in principle, but in order to make it efficient, you need a lot of work, a lot of ideas. So basically, nowadays, modern molecular dynamics tools, codes are extremely efficient, are parallel and highly scalable. So this required a lot of work, well, more than 3 decades of work by, well, not really hundreds, but I would better say thousands of people. So nowadays, the molecular dynamics tool that we have in our hands are extremely powerful. We have to understand, well, the first thing we're going to understand in this first part of my talk is how powerful they are. So why is it necessary to have powerful codes? Because if I want to simulate a realistic system, for example, a realistic biomolecule, I have to cope with 3 different problems, 3 different codes. So one is that I would like to simulate large and inhomogeneous systems. Here, this is the image of a membrane channel, for example. So this is like 300,000 atoms easily. Well, then I have a problem with the time scale. This is the one on which I will dedicate a little bit of time in this presentation. And the other issue is the accuracy. One would like to have from these simulations a quantitative answer, not only a nice movie or a qualitative description, but really something that helps you to understand how, for example, a biomolecule is working and eventually adapt it in such a way that you can, for instance, control its behavior, right? So here, in order to do this, you need accuracy. So here you have to choose the correct level of accuracy for performing your simulations. Here, in this overview, I will simply start from that because this is actually the key point which will then determine how much time we can simulate. So which level of accuracy of description should one choose? So this clearly strongly depends on what you want to simulate. So, well, let's say the cheap option is using classical potentials. If you use a classical potentials, of course, you will not be able to describe a chemical reaction, but you can really do a lot. You can already study how, for example, a protein works, how a typical protein, not an enzyme, is able to perform its task. So here there are no quantum effects on atomic motion, no electrons, no chemistry. Well, what we call a force field is a potential energy function and a set of parameters which enter in its definition. It is designed to reproduce the molecular geometry and some selected properties of the molecule. Also, in order to develop these force fields, this required really a lot of work and a lot of insight. It's now almost a 4 decades old story. So here, just to give you an idea, so this is the functional form of a potential energy function for performing biomolecular simulations. You have a bond term, an angle term, this one. Then we have a dihedral term, which describes the interaction between 4 successive atoms. And then, and then we have the non-bonded part, which is actually the rate limiting part. So here, computing the first part is somehow cheap because it involves only the few atoms which are close by. Instead, computing the second part is what makes the computation expensive. Okay, so here, then you have another option. Let's say that you want to describe a chemical reaction, then you have to deal with the electrons. So if you want to deal with the electrons, you have to go and chat a little bit with Mr. Schrödinger and use his well-known equations. Okay, then in principle, you would like not only to solve the electronic structure for a given configuration, but also do dynamics using the forces that are derived from your electronic structure. And then you want to use at the same time the Schrödinger equation and the Newton's equation. And the manner of using these things together was actually first developed and invented here in SISSA by Carparinello in their famous 85 work. Okay, so this approach is great because at variance with a classical force field like the one that I described in my previous slides, it does not require any fitting, no free parameters. Everything comes from first principle, but of course you pay a price. And this price is that you basically, it is orders and orders of magnitude more expensive than the classical force field. So orders and orders of magnitude. Okay, so here, now this is an important slide because it gives you a little bit an idea of what we can afford simulating nowadays. So let's start with the classical force field. So let's make an example, simulation of HIV protease with a classical potential. So this system has approximately 50,000 atoms. Okay, so each atom interacts approximately with 100 atoms, let's say its neighbors. So in order to compute the forces in a given configuration, I need to do something that basically I need to compute forces between 50,000 times 100 atoms. So this, in order to perform this number of calculation, an ordinary CPU, a single ordinary CPU needs to do basically this can be done in approximately 0.2 seconds. So in 0.2 seconds, I compute the forces, all the forces on this system, and then I am able to move my system for one step. So here, basically, in one day, I can move the system. Well, here, basically, you can read the calculation, it's very simple, but basically, 500,000 times. I can move it 500,000 times. So this means that I move my system 500,000 times by a time step. This time step is, if you know a little bit about molecular dynamics, is 1 femtosecond, which means that in one day, I will be able to basically simulate 1 nanosecond. 1 nanosecond in one day. One would say, so this is an enormous technological success if you compare to what we were able to simulate decades ago, but it's only 1 nanosecond in one day, no? Okay, so here, this is what we can do with classical potentials. If I want to use a Carparinello or a quantum potential in which electrons are treated explicitly, what I can actually simulate is something much smaller. I can simulate a much smaller system, and instead of 10 to the minus 9 seconds, I can simulate only 10 to the minus 11 seconds. Well, of course, I can use something which is much better than one single CPU, and people across the world have built large computers. In particular, David Shaw in New York City has built a computer which is able to do only molecular dynamics of proteins. So this gives you an idea of how important this task is. So he has invested an enormous amount of money. So this is the money that he has invested, $100 million of his own money. So this guy decided to invest his own money in order to make this thing work. And he has built an hardware which is able to do only this. So of course it is much faster than our computers, of course. No? So what can you do with that? So with that, you can simulate 17 microseconds per day on a system more or less of the size that I mentioned in my previous slide, so 50,000 atoms. Okay, so this is an enormous technological advantage, and in fact, these guys are obtaining amazingly good results using these fantastic computers. But how far can we go with these numbers? Okay, so let's say that I am rich and therefore I have this computer, so I can simulate 10 to the minus 3 seconds in 1 month, 1 milliseconds. So this number 10 years ago would have looked like a miracle, science fiction. So let's say that here with my resources, I am able to simulate instead 10 to the minus 5 seconds in a month. So 2 orders of magnitude less, but still quite a lot. But now, okay, so the typical time scales of interesting transitions are typically orders of magnitude longer. So even protein folding, 1 millisecond, there are many proteins which, in order to fold, take almost a second. So this means that we are, even if I have this enormously powerful computer, I will not be able really to see even a single folding event in many proteins of great interest. The situation is even worse if I want to simulate phase transitions. Phase transitions happen on mesoscopic timescales. We are used to see phase transition happening in our real world time, which means seconds, hours. So how can I simulate a phase transition if I am only able to simulate 1 millisecond? I have a problem. Ab initio is even worse. Chemical reactions, also chemical reactions. There are many chemical reactions which happen on the time scales of minutes, hours, days. So these things are clearly out of reach for direct molecular dynamics simulations. Okay, however, even with the moderate computational resources, we can do something. I'm going to show you 2 examples which are, well, both not from my lab. So this is a simulation of the folding of a knotted protein of 86 amino acids, which in real life folds in approximately 0.1 seconds. So 100 times longer than what it is possible to simulate with the big computer which I mentioned. So this is the folding trajectory, which was obtained well, in the lab of Pietro Faccioli in Trento. So it's possible to obtain this thing, and it's possible to obtain many folding trajectories like that. First example. So this is a second example, a phase transition in a zeolite. So this system, in order to form, to observe a phase transition like that, so this is something which in real life happens on the time scale of minutes, if I remember correctly. So here, already more than 10 years ago, it was possible to obtain, observe systematically and study systematically many phase transitions of this system with moderate computational resources. So here you see a trajectory in which the system really jumps from one phase to another. How is this possible? How is this possible? How is this possible? Well, the key for making this possible are enhanced sampling techniques. Here, of course, in the 20 minutes that are left in my presentation, I don't have really a chance to give you even an overview of everything that is around, but at least I would like to leave you with the idea of what is the gist, what is the main thing that make this miracle possible. Okay, so how is it possible to pass from well, a simulation that is 10 to the minus 8 seconds to get information on a process which would occur in on the scale of seconds? How is it possible? All right. Well, let's try to give a close look to the systems which we already mentioned. So the idea is to understand a little bit what's going on. We should first of all find a function of the coordinates which is likely, based on our chemical and physical intuition, to take very different values in the different states. We call this function S of x. So here, for example, in the case of phase transition, a good function is the box shape. Here you see that in this transition, the box, simulation box, changes shape. Here in a chemical reaction, the bonding pattern, here you see that this atoms form different bonds in the different states. Here, in the case of protein foldings, I can choose many, the fraction of alpha helix, the number of contacts, the gyration radius. So I have to find a function which basically takes different values in my different states. Okay, now, how would this function behave as a function of time? Okay, so this is an example. Here, this is a very simple system. Here I have simulation time, and here I have my collective variable. Here you see that I have a fluctuation in one state, and then a jump to another state. Okay, so here, so this is the time before observing the reaction. So this is the time which is of the order, for example, of seconds or of hours or of days. And this is the reaction time. Here you cannot even almost see it, but it's actually much, much shorter than this time before the reaction. When we say that protein folding of this knotted protein occurs on the time scale of 0.1 second, we are actually talking about the time before the reaction, not about the time that the reaction takes to happen. So this difference, this subtle difference between these two things is actually our handle, where what we use in order to develop our enhanced sampling techniques. So how do we do it? So in a molecular dynamic simulation of a chemical reaction, a phase transition, the system actually spends most of the time oscillating in a local minimum. So this is actually the key point. Only rarely it performs a jump to another state. The time required to perform the jump is short. It's very, very short. For example, in a chemical reaction, you can have a waiting time of 1 hour, but then the time that the reaction occurs to really happen as soon as it starts happening can be 1 picosecond, 10 to the minus 12 seconds. So 1 hour of waiting time with nothing happening, and then 10 to the minus 12 second, everything happens all of a sudden. So the key trick, which is at the basis of all the enhanced sampling technique, is finding a manner to simulate only the jump. So this is actually the key trick. Then all the different methods work in different manners, but this is the key trick of the business. Okay, how do I do this? Here I can do it well in many different manners. Here, as I said, in order really to give you even a hint on all the different manners, I would take hours. But okay, one idea is to pool the collective variable S. So once you have found that your system is nicely described by a specific order parameter, for example, something which describes the order of your structure, then you can simply add a spring on this specific variable and simply pull it, like exactly you would pull a spring and force this collective variable to change. So this is the idea which is at the basis of thermodynamic integration, which is a very well-known method and also steers molecular dynamics. Then you can do something which is more rigorous, which is, well, I like to call it important sampling on reactive trajectory. Exactly like you do important sampling on configurations when you do Monte Carlo, you can actually devise a sampling scheme which preferentially sample only reactive trajectories. So the most famous approach in the field is transition path sampling, but actually there are other methods which are also related with this idea of simulating only the reactive part of the trajectory, for example, the finite temperature string, the nudge elastic band in the framework of chemistry. Then, okay, another idea which I will actually describe in a little bit more detail is the idea of flattening the free energy. Here, I will spend the next few slides on that, so I'm not going to tell you anything on that at the moment. And then another idea is to rise the temperature. So of course, if you rise the temperature, your rare event become less rare, but you cannot simply trivially rise the temperature of everything, because otherwise you simply melt the system and transform it into an ideal gas, which is not very interesting. No? So you have to rise the temperature in a smart manner, and you can do it in many different manners. One manner is temperature accelerated molecular dynamics, where you rise the temperature only of the collective variable, only of S, or you can rise only the temperature of the solute. It's called solute temperature, really. So all of these approaches have their own history, their own advantages and disadvantages. Now I'm going to spend the last part of my presentation in describing a little bit more what it means flattening the free energy. So what is the free energy, first of all? So here, this is the trajectory that we were looking at before. From this trajectory, what you can compute is the probability density. You can estimate the probability density as a function of your collective variable S, and it will look like that. You will have a maximum here and another maximum here. This maximum corresponds to this state, and this maximum corresponds to this state here. So from the probability density, you compute, you define your free energy. Your free energy, F, as a function of S, is just minus kBt, the logarithm of the probability density. So here, if this is the probability density, this is the free energy. So a minimum in the free energy corresponds to a maximum in the probability density. And so in this specific system, you will have a free energy which looks exactly like that, with two separate minima. So here, what does it mean flattening the free energy? So let's say that I start with this free energy here. If I do my simulation on a system with this free energy, I observe a trajectory like that. Long waiting time, and then only few transitions in this time that I am able to simulate. Then they say that I add to this free energy an external bias. So the external bias here is, in this specific example, which I'm going to show, this weird form, which is a sum of two exponentials. So the sum of the free energy plus my external bias will be this red line here. So you see it's flat. Now, if I simulate my system under the action of my normal potential, which has this free energy here, plus this external bias, what I get is this trajectory here. It's the same system, but now you see that the system is going up and down again and again and again. So this thing is actually making exactly what I was telling you before. So adding this bias allows me to simulate only the jump, only the jumps. But now, so therefore, instead of wasting all my simulation time in waiting for the system to vibrate, and then for a very rare event which happens only from time to time, I force the system to go back and forth again and again, and this allows me to observe many, many times the relevant transition. Okay, so here this is great. This was actually invented in the 70s, and it works great, but well, we have a problem, and the problem is who gives us this fantastic bias potential which is able to flatten at least approximately my free energy, because of course when I start my simulation I don't know how the free energy looks like, right? So this.
So Alessandro, sorry, you have 4 more minutes.
Yeah, why? Pardon?
4 more minutes.
4 more minutes, yes, but I am almost ready.
Okay.
So how can one find a bias potential B of S which is approximately equal to minus the free energy, and which therefore when added to the free energy gives me something which is approximately flat. So here, so I have many, many options which were all used again and again and again across the years. Well, first of all is using my chemical or physical knowledge to invent a correct bias potential. Option number 1. Option number 2, simulations with simpler potentials. I simulate it with a cheaper potential, I compute the free energy, and then I define my bias using this previous simulation. So the other option is an automatic procedure. I'm going in the last 3 minutes give you some hints on a possible automatic procedures, which is called metadynamics. So in metadynamics, the bias potential flattening the free energy is built iteratively by an approach which consists in a loop described in these 2 lines here. You basically put a small Gaussian and then you evolve the system for a short while under the dynamics of the small Gaussian plus the free energy. So first you add 1 Gaussian and you go there, then you add another one and you go there, you go there, you go there, and you go there. So basically this idea of adding from time to time a small Gaussian actually is a procedure which allows you to iteratively flatten your free energy and iteratively build a bias which compensates your underlying free energy. So more rigorously, you choose a collective variable S of X and you bias the dynamics with a history dependent potential which has this form here. Here we have an example of the free energy, which is at the beginning unknown, plus the sum of these Gaussians, which iteratively become more and more flat. So here I have the sum of my free energy plus the bias potential defined by this equation, which is flat, which actually means that for large time, my bias potential defined by this equation here is an approximation of minus the free energy. Okay, so here is an example of normal molecular dynamics and metadynamics applied on a system undergoing a chemical reaction, which in normal Carparinello molecular dynamics would be absolutely impossible to simulate. And here, well, and this is, well, really I am finishing, yeah, maybe with this movie where still you see nothing happening for the moment, but now all of a sudden you see that the guy is able finally to do some stuff and performing a sequence of transitions in which the system actually jumps from one free energy minimum to the next, and well, exploring the conformational space in a very efficient manner. So here you see that now the system is actually performing a transition to a molecule which is called naphthalene. So it's almost gone, and now it goes to naphthalene. We wait another second. Yes, we wait. Okay, now it's gone. Now it's naphthalene. And with this, okay, well, here, of course, I could show you other examples of systems, more interesting systems on which we have applied these things. We have studied many systems with these approaches. For example, the folding free energy landscapes of a protein, or the free energy landscape associated to the nucleation of fibrils. Here I could show you many examples, but actually I would like to close with a slide where I show what, in my opinion, are the open problems which are still at the center of the research, well, not only for this specific method, but for many methods. So the first important problem is how to find automatically the best collective variables. Here if you don't, if your system is very complicated, it's very difficult to know in advance what is the best variable on which you should add your bias or pull your simulation or rise your temperature. So this is the first important problem. A second important problem is that using these powerful and unsampling approaches, you actually observe many transitions. You are actually able to explore very complex conformational landscape. And an open research topic at the moment is how can I analyze automatically these complex conformational landscapes? So both these two problems actually call for a combined approach using machine learning, artificial intelligence, and data science tools. So this is basically, well, the reason why I personally am more and more trying to use approaches from this field, simply because they give you tools which allow you to address this problem, which otherwise with classical method would be more difficult to address. Well, with this, I'm finishing my presentation, and thank you very much for your kind attention.
Thank you very much, Alessandro, for the very nice talk. And so as usual, now we are open to questions. Let's see if I don't know if in chat there is no. Okay, there is, yeah, there is.
Hi, can I ask a question?
Yeah, sure. Who is he?
I'm Uriel Morrison.
Yeah, so sorry. Yeah, just for the question, please raise your hand. Otherwise, I cannot see.
Sorry. I
no
don't
problem.
know.
No problem. Now go on, please.
Okay. Thank you, Alessandro. It was very interesting, and this is a very naive question. I don't know much about these methods, but it's like two questions in one. One is, does the metadynamics preserves the statistics, like ensemble statistics, like canonical ensemble, or, I mean, are you violating that, or is it important to preserve it or not?
So if I can answer immediately, this is a very important question. It is not, in the sense that it is a dynamics which basically adds a history-dependent potential to your system. Therefore, the probability distribution of the system is, of course, not anymore canonical. But during the years, many people have done a lot of work, and it is now, this probability distribution is actually now very well understood. And even if it's not canonical in the initial variable of the system, basically people are now able to extract the original probability distribution, original canonical distribution, as doing basically a post-processing of the biased probability distribution. So the answer is yes, this dynamics perturbs the probability distribution, but there are results which in some conditions are exact results, which allow you really to basically compute back the correct probability distribution from your metadynamics trajectory.
Very nice. Okay, this answered my second question, which was how to obtain the free energy from this method. So
well, basically, from this method, is the free energy is, naively speaking, is minus the bias. More rigorously speaking, is minus the time average of the bias. So basically, in metadynamics, the bias becomes a dynamic variable itself. Is like having another dynamic variable. This dynamic variable is a field because it's a function, and in order to obtain an estimate of the free energy, you take the time average of this bias. So this is also not a rigorous answer, because a rigorous answer would require having a blackboard and much more time, but just to give an idea, you take the time average of the bias.
Okay, so this is true even if the statistic is not preserved.
Even if the statistic is not preserved, this is rigorously true if the dynamics in the collective variable is adiabatically separated dynamics, your other variables, or in the limit of infinitely slow depositions. There are technical details.
Thank you very much.
Sorry, Alessandro, we have a few questions.
Yeah.
One from Dennis Mteremko. What collective variables did you use for your study?
Well, an enormous amount of collective variables. The thing is that this is an art. So if you go to Plumed, Plumed is the plugin which implements all the rare event methods, including metadynamics. You will find a list, I think, of 50 collective variables. But just to give you an idea, you can use like coordination numbers which describe the bonding pattern. You can use the energy. The energy is total energy is a very good collective variable. You can use the radial distribution function. You can use the stress. You can use whatever you want. Any possible function, explicit function of your coordinates can be a good collective variable for studying your system. So this is where the fun of our works is somehow, no? Is choosing the correct collective variable for any specific problem.
Yes, there is another question from Lorenzo Pallante. He's asking, in your opinion, what is the best method to define a realistic transition path between two states? And could you recommend some tools or software for biological systems?
Okay, so here if you really want to define the pathway, the transition pathway in a rigorous manner, in my opinion, the correct tool is transition path sampling. The approach developed by in the group of David Chandler, Del Lago, and so on. So here, so this thing is something which is totally agnostic. It doesn't require choosing a collective variable. It simply does important sampling in the trajectory space with no prejudice, and then you analyze a posteriori these reactive trajectories. This group, they develop their own tools. If you browse on the web for their names, you will find tools which allow you to do this thing cleanly. So this method, on the other hand, doesn't allow you really to explore your configurational space like we are doing in metadynamics. It's more a tool which, if you know reactants and products, where the system is going and coming, this allows you really to analyze rigorously how the system is performing the transition.
Okay, so maybe one last question from Alice Romeo. She is asking, what's the difference with Gaussian accelerated MD, in which you automatically add the boost potential without predefining collective variables?
Well, I think it's so any method. So I don't think that what you say is possible, in the sense that what you can do is to add a Gaussian acting on all the coordinates, for example, all the dihedrals, like it is, for example, done in not adaptive force bias, in flooding, in so I don't remember the name of the approach, but okay. So, but this approach does not allow you to compute the free energy, because the bias, if it's acting on too many variables, does not really converge, because the size of the space which you are exploring depends on the number of variables on which you are putting the bias. So if you are biasing, if you are simply putting a Gaussian on the coordinates, then you are biasing a space which has a dimension 30,000, and you will never be able to fill with Gaussian a 30,000 dimensional space.
Okay, yeah, sorry, I think we stop here. Thank you.
Well, of course, let me say that if you have other questions, you are more than welcome.
Yes, exactly.
To me privately in email, or I don't know if we have a public chat associated with this workshop. Okay. So in any case, my mail was on my first slide. You can see it on the recording if you want.
Okay. Thank you very much, Alessandro, again. So we move to the next speaker, and I have to ask you to accept the invitation to the breakout room.
Alessandra, buongiorno. Dovrebbe aver ricevuto un invito per la breakout room.
paraplegia.
Hello, sir, can you hear me? Professor Rodriguez, can you hear me?
Yes.
Okay, you can start the test, please.
Okay, shall I start sharing my video?
Yes.
Can you see me?
Okay.
And then my screen. Can you see that?
Yes. Can you activate the full screen?
Yeah.
mode? Okay.
Is it
Right.
working?
Yes.
Okay.
Okay, so we are already streaming, so we are ready to start. I'm going to start recording. I kindly ask the moderator to tell me if we can start in 7 minutes.
Hi, can you hear me?
Yes.
Yeah, I think it's fine. So the only thing is that the first talk will be in the separate room. So if the third and fourth speakers are already here, so Ali, I think will join later.
Yes.
If Angelo is already here, maybe we can try his screen sharing.
Oh, yes, because Professor Micheletti does not want to be recorded. Okay, right. But we expect some more participants, right? We have 30 participants now.
Yeah. But I mean, if so now we are not in streaming, right?
Yes, we are in streaming because we are streaming all the day.
I see.
In a single block on YouTube.
Okay, so if Angelo is here, maybe we can test his connection.
Okay.
Sharing.
Let me check. No, he's not in yet.
Okay, so I think we can wait a few minutes, and then around 2, maybe if Angelo and Ali didn't show up yet, we will move to the other room and then start with the talk of Christian Micheletti.
Okay, so I'm back in a couple of minutes. I have another assistant, sorry.
Okay.
Okay, Professor Rosa is in.
Ciao Christian. Ciao Angelo. So we can test your screen sharing, Angelo, so that we are sure there will be no problem afterwards.
You are muted.
Yes. It should be this one. Can you see it?
Yes.
Does it work, Giovanni?
Yes, yes.
Stop. Can I do stop sharing?
Yes.
Yeah.
Of course, later it will not work.
Professor Bussi.
Yes.
Le rispondo subito. Man mano che la gente arriva, perché anche prima è successo, ci sono tanti ritardatari, io li dirotto nella breakout room.
Ok, perfetto. Quindi A questo punto, visto che Ali, non credo che sia, se ho capito bene Angelo, Ali arriverà più tardi, un po' più tardi,
Ali
giusto?
cannot join now.
Ok, quindi forse possiamo direttamente aprire la breakout room e fare la prova di proiezione con Christian Micheletti.
Ok.
Avrei aspettato se ci fosse stato qualche problema a dirottare i partecipanti, ma se è automatico possiamo già aprirla, penso.
No, li dirotto io manualmente.
Ok.
Man mano che arrivano. Ok. Vado. Please all join the breakout room.
Mi raccomando, no, cerca di scrivere non troppo piccolo perché dobbiamo... Ecco, guarda come si vede, no? Così. Questo è... No, dico, questo è la bagno. Questo è quanto, allora lo vedranno, possono vederlo. Grande, no? Sì.
We can see you and hear you.
Professor Potzer, good afternoon. This is ICTS.
Tarek, I cannot hear you.
Can you hear me?
You hear me now? Is that me or? Can you hear me now? This is Tarek. Can you hear me now?
Yes.
It's a problem of my system. Here we are. So, can you hear me now? Can you hear me now? Okay, sorry guys, there was a problem with my headset. Okay, good, perfect. Now I suppose I can hear most of you. Tarek? Yeah. Okay, perfect. Yes, perfectly. Okay, sorry for that. Okay, that's great. So
Professor Potts, excuse me, can you hear me?
Yes, I do. You are?
ICTP, ICTS, IT services.
Okay, yes, I can hear you very well.
So, are you the host? No.
No, I got some host password in case, but I don't know how really it will work. So, because I think we are supposed to record.
Just a second, let me claim the host and promote you.
Okay.
Sorry, just a second. I apologize, we have several activities at the same time and lack
No
of
worries,
people.
I completely understand. So just if I may, okay, I can claim host.
Yes, but you don't have the host key.
Yes, I do actually. This,
You have it?
Yes, I have it.
Okay, try to do it by yourself.
Okay, I'm going to do that.
Perhaps you received some instructions with the host key about recording.
Actually not, so that's just the problem. So now I am host and I can record. And I just want to know what shall I do now?
So if you have to record, then you'll have to put the files on a Google Drive to link shared with you. We will provide you this link and we will kindly ask you to upload there the files. And after this, we'll provide to upload them on YouTube.
Okay, normally when I type record, it will be record, I have to set up where it's going to
On local computer. Please never record on cloud.
Yep, perfect. Okay, and then we will discuss later on.
So you confirm me that you did not receive any Google Drive shared link.
At the time, no.
Okay, it will be provided to you as soon as possible.
Yeah, no worries. I mean, so let me record it and then we can just, yeah, okay.
Okay, very well.
Good. Coffee. So everybody can hear me. We are roughly 12 people. That is good. So the first question is quite simple. Does any one of you ever had atmospheric chemistry before?
For me, it's not. It's
totally new.
New, yeah, it's new for me.
For most of you, it's totally new. You had physics, I suppose, of the atmosphere, a few things, so fluid dynamics, I suppose. But atmospheric chemistry, nothing at all. Is that correct?
Yeah.
Good. I do apologize because I see all of you nodding. That's just not again, I should say.
I think I read some bits of atmospheric chemistry.
Atmospheric chemistry is everything. Okay, good. So let's start with the recording. So
no. Forse registra lui, che dovevo mandare il Google Drive e lui mi metterà dentro il file.
Okay, so in a second I need to move. Yeah, okay, good. So let me check the last things before we start. Oh, sorry, you know this COVID is killing us, so it's the first time.
No,
last year we did all remotely. Record, of course, for most of you, it's also recorded after recording. So, other question is is it working? Is everybody happy with that? So let's get that. Is everybody having a problem? getting the information. Okay, good. So, because of course,
Well, there are 2 breakout rooms. I don't know why. Okay, let me do this. I'm gonna close all the rooms and reassign. Pronto?
Chemistry, some actually, that means actually we try to squeeze all the chemistry that we need to know in only this semester. That means that actually we will have like 7 lectures.
Non so cosa dirti, Vittoria. Non ci sono altri modi. Cioè, tutto.
Okay, now I guess everyone is in the main room.
Not yet. Not yet. There are still some people.
I think that there were 2 rooms by mistake. It would be important to have a single breakout room. Otherwise, we make.
I'm gonna recreate the room and I'm gonna open it. Okay, you should receive.
So that's so cannot start.
Alex, your microphone is off. Okay, great. Yeah, I think the transition was very smooth. Now everyone is in the room, so I think you can continue.
Can you see my slides? Yes?
Yes.
Okay, perfect. So I was saying that indeed what we want is that the computer made for us this task, that it's to identify the groups in our data. And we want to do it even if the data is in high dimensions, and if the clusters, our groups, are odd-shaped. We will see later what it means. But before going there, I want to say why clustering. We are in a atomistic people works. Well, I will not say event, let's say, and why we are talking about clusters. Let me show you 3 cases that are not all of them, but maybe kind of general. One case is the analysis of a molecular simulation. In this case, our data, it came, this is a protein. So it's a converged sampling of this protein that has something like 10,000, 100,000 coordinates. And from this simulation, what we usually want is to obtain the kind of information that it's in the right, that it's exactly the conformers. To obtain these conformers can be done by clustering with few knowledge of what's going on in the protein. Another test case is the chemical databases. For instance, I have 4, well, this is 4, but usually it's millions of compounds databases. And when I apply clustering, I apply it inside a pipeline that allows me to reduce the number of compounds that I want to test against a given target, for instance, in the case of drug databases. Another case that it also has biological applications, it's case when analyzing sequences. This is a protein, but it can be also applied to RNA, DNA sequences, where from just the sequences, I can group them. And these groups can be useful. For instance, in the right, I have shown an example from a paper in which they perform a clustering in protein sequences in such a way that they identify which clusters are more likely to have proteins that have a given property. In this case, were mood killers, something like that. I don't remember exactly. But the idea is you cluster all the proteins and you have in the underlying hypothesis that similar proteins that belong to the same cluster, would have a high probability of having same properties. So when clustering, I took this slide from my lessons in clustering. You first start from data, and then you usually perform some kind of feature transformation that allows your data to adapt better to your target. And then you perform the clustering. Usually then you validate the clustering and interpret them. And usually from this interpretation, it's where the knowledge appears. But this project this pipeline has some feedback, because when validating, you can came back to perform a different clustering, or even came back to a different feature transformation. But let's start from the beginning for the data samples. One thing that you have to have in mind is how many objects are in my data set. Why? One thing is the computational power. Let's say if you have millions, billions of data points, there are some kinds of analysis that you cannot afford. Instead, if you have hundreds, it's much easier. But in the other hand, one has to take into account that for some methods, there is a kind of statistical sampling needed. You can think about converged molecular dynamics. You need many points for converged molecular dynamic simulation. So also that it's something that you have to take into account at the moment that you decide your clustering method. More important even, it's how is your data described. For instance, in the case of the atomic coordinates that I put at the beginning, your features are the atomic coordinates, real numbers, right? In the case of the chemical library, your data, your features are the structures, some kind of descriptors, but also the 3D structures, both 2D and 3D structures. In the case of sequences, usually you just have the letters. Okay. Once you have cleared this data, you have to deal with the feature transformation, and feature transformation usually implies 3 steps, or may imply 3 steps. One is feature selection, one is dimensionality reduction, and the other one is distance computing. Let's start for feature selection. What I mean with feature selection. Well, here I just explaining you what is that. There are many automatic techniques, but it goes far away from the purpose of this talk. And the question is that, for instance, in the case of atomic coordinates, I can wonder if all of them are relevant for my purpose. If I'm interested in the folding of a protein, probably I shouldn't include the water molecules. Maybe yes, but probably not. The same occurs with the side chains. I mean, for instance, people working on folding, again, usually just include the main chain atoms. But if you are interested in other kind of analysis, probably you need to include the side chains, or even you can need to introduce the water molecules. Ali would explain us later why. In the case of chemical databases, the question is that which descriptors are relevant for my problem, and this is much more difficult to get because, and there is a lot of expertise. For instance, you can think about the log P, measure of solubility, the pharmacophore profile, that it's a kind of 3D descriptor, connectivity indexes that depends on the topology of your molecules, and so on so far. But there are so many that you usually need automatic tools for choosing them. In the case of sequences, what may happen is that you have to clean somehow your sequences in order that avoid the introduction of not significant segments, mostly, let's say, when you have monomers against dimers or something like that, it's probably you just need to take into account some monomers. Regarding the dimensionality reduction, it's a field by itself. I'm just going to introduce some hints, but I don't want to do the talk in dimensionality reduction. Dimensionality reduction, it's okay. Independent of the data that I have, I can try to reduce their features to few features. In this case, I have just 2 features. And this reduction can be meaningful or not meaningful, depending on the methods and on the system. There are many methods that allows us to use to perform dimensionality reduction. This scheme came from an already kind of old review from 2009. There are much more methods nowadays, but well, I would not explain them, but just to say that it's still an open problem. And why? Because it can be really easy or it can be almost impossible. In the case that you see in the screen, you can see that this line is kind of easily transformed in 1D. And this can be done by principal component analysis. That is a technique based on the covariance matrix. And it's a technique that works in, let's say, linear environments, let's say, in hyperplanes. However, if you have something like this 3D point distribution, with PCA, you cannot reduce the dimensionality from 3 to 2. It will not work. And it will not work because this surface is twisted and it's kind of topologically complex. However, people have been working on that. There are methods that work pretty well on nonlinear projections. But the question, it can be even much more complex. If you see these distributions, and I say that it's just a 2D distribution of points, but this distribution can be summarized in a line if you want. But it's difficult, it's one-dimensionally, but to put it in a single line, it's topologically impossible, okay? So sometimes the projections are not easy to do. And this can be seen in two cases. For instance, this is a simulation from the results from an old simulation that I made 9 years ago. And in this case, PCA works fantastically, and projecting the coordinates of the backbone in 2D with PCA allows us to identify two free energy minima. That allows us to identify two conformers of this mini protein. It's a peptide that folds. Okay. Instead, in the case of Bilin headpiece, we try also to project with isomap the coordinates of this. It's bigger protein. It's 32 residues. And the question is that by projecting with isomap, you can see that, okay, the ground truth is the color code that tell us the degree of folding. And you can see that the folded and unfolded points, configurations are mixed. And this happens because isomap is not able to project in 2 dimensions all the complexities that have the configurations of the folding trajectory. Okay. Therefore, in dimensionality reduction, we try to project in 2 or 3 dimensions. But okay, if it works, it's perfect. You can almost finish there. And even if it doesn't work in 2-3D, if you project in, let's say, 8 dimensions, probably it will simplify the analysis a posteriori. However, if we don't do it properly, we can have an important information loss. And if the data set lives in a complex manifold, it's not easy. It can be difficult or almost not even possible to perform meaningful projections. The last point of feature transformation that I want to comment is distance computing. And the question is that most of the clustering methods rely in some kind of quantification of similarities. It means that for clustering, we need to quantify how similar or dissimilar are 2 points, okay, 2 data points. In the case of proteins would be configurations. In the case of the chemical database would be compounds. In the case of sequences would be each sequence, okay? These similarities for thinking about clustering can be interpreted as distances. And that's important because it allows us to reasoning in clustering terms. And the question is that the lower the distance, the more similar are 2 points. Okay? 2 points that are in top of the other would be identical. 2 points that are really far away would be really, really different. But our definition of distance depends also on the data type that we are dealing with. There are some general distances that are pretty used, like Minkowski distance, that it's a generalization of the Euclidean distance. You can see that with with P equal to 2, this recovers the Euclidean distance, or the cosine distance that just take into account the angle between the vectors describing our data. There are distances that are defined for molecular systems like the RMSD, or the diagonal distance that defines the Euclidean distance taking into account the periodicities of the diagonal angles. Also for binary molecular descriptors, we have the Jacquard distance. We have the Hamming distance that allows us to compare 2 protein sequences once they are aligned. The Hamming distance is increased by 1 if the amino acids are not the same, or set to 0 if they are the same. So there are many, many distances. I just put here some of the most used in our field. And finally, we can start talking about clustering, real clustering. Okay. As I told you before, well, the clustering is relatively easy for humans, but not always it's easy. I mean, now I'm going to show you many clusters obtained from this distribution of points. One would be this, that it's kind of trivial, but you can argue that all of them could be correct. So at the end, the clustering result depends a lot in our definition of what is a cluster. And of course, we have to define a cluster according with our target when we perform clustering. And this is the reason, one of the reasons for which there are so many clustering methods. Okay. However, they can be somehow summarized in 3 types. One is flat clustering, that it performs just a hard partition of the data. It's what we have all in mind when we talk about clusters. It just assigns each data point to a group. And I put here some widely used methods for perform this flat clustering. Fuzzy clustering is a bit more complex because the assignment to a group, it's not univocal, but it's you give to each point a degree of membership to each cluster. Here there are other methods that employ fuzzy clustering. And finally, there is hierarchical clustering that generates a tree instead of a single partition. And this tree, you can see a kind of hierarchy. I mean, for people that have seen, let's say, nature programs over, let's say, the families in the animals or the taxonomies. Taxonomies are kind of hierarchical clustering. Okay. let's see what are these points, let's say. In flat clustering, everything is easy. Let's say I have this partition where points in red belong to cluster 1, points in yellow belongs to cluster 2, points in green to cluster 3, and so on. In fuzzy clustering, I have a kind of palette of colors, right? I would say that this point, the point that I'm signing with the arrow, has a 90% of degree to cluster 1, a 9% of degree of membership to the cluster 2, and a very, very few degrees of membership to other clusters. This other point is almost in the half between these two clusters, so has a degree of membership to each of them, and also to the third one. This one is kind of in the middle of many, and so on so far. Okay. Hierarchical clustering makes something more complicated, but by generating a kind of hierarchy, you can recover the clustering structure. Okay. But this clustering structure, it's difficult. It may be not easy to transform in flat clustering. For instance, in this case, the two clusters are easily recovered, but for recovering the 5 clusters, I have to do a kind of strange cut, right? However, there are some there are other differences in our clustering. For instance, instead of inside the flat clustering category, I would like to remark 2 clustering methods. I would say the partition methods that allows you to put together points that are similar, while density-based methods allows you to follow the profile, the priority profile of the points distributed in your data. The difference is that, I mean, there is not a correct clustering. It depends on what is the utility that you are going to use this clustering for. According to your target, you may want to use a partition method or a density-based method. Okay. For instance, a typical case of k-means clustering of partition methods, it's k-means clustering. It's probably the most used clustering method employed, and it works pretty easy. Let me explain you qualitatively. The first thing that you do, for instance, in this dataset, is to randomly pick k-centers. You have to decide how many clusters you want in this method. You randomly pick them, and then you assign each point in your data to its nearest center. Once you have this clustering partition, you can recompute the centers that somehow will move. Okay. And then you can iterate. And by iterating, the clusters, somehow you find an optimal Delaunay partition of your data. The only thing is that this method depends, of course, in the initialization. In this case, you can see that for this initialization, there are 2 clusters that are divided by 2, while there are 2 clusters that are merged due to the fact that my initialization was not perfect. Okay. Different method, it's based on density. I would want to explain you. It's density-based clustering. And in this case, like in all density-based clustering, the first thing that you have to do is to compute the density for each point. And the density is nothing else than a quantification of the number of points in the neighborhood of a given data point. Once you have computed the density, that it's kind of trivial compute, you compute the distance from the nearest point with higher density. and we call it delta. And then you plot this delta as function of density. And what happens is that the points that are outliers in this graph happen to be centers of putative clusters. By picking these centers, once you have picked them, you can try to assign each point, for instance, this red point, by following the profiling density and arriving to a given center. Once you arrive to a given center, you assign all these points to the same cluster. Oops, sorry. Therefore, if you do it for all the points, you finish having a full partition. Okay. Okay, I don't want to explain you much more methods of clustering, also because I don't know how much time I have, Giovanni.
Yeah, a couple of minutes.
Okay, so I just want to finish with some applications. Okay, let's back to the applications that I told you before. Imagine that you want to cluster the Billing headpiece. By using a mix of density methods with hierarchical clustering, you can obtain something like that. That translates in this free energy profile. Okay. This free energy profile, you can obtain it because you have the densities. By using that, then feeding a Markov state model with this output, you obtain a 5-stage Markov model for Billing headpiece. Okay. Another application, it's in the field of chemical diversity. Let's say these people just make a hierarchical clustering based on descriptors of the compounds mixed with the fungi. They are fungi, they came from mushrooms, mixed with the genetic analysis of the mushrooms, and they managed to obtain this nice hierarchy that explains how the diversity on these products was born. Finally, in the case of protein families, we work with the Pua family, and we obtain that this clan, that it's a clan, the clan of Pua, can be divided in many families. According the histogram, the clustering results are in the left, while the ground truth is in the down. And you see that even not exactly, somehow the clusters, let me show it, let's say, all this part of the dendrogram that comes from the cluster reproduces this family. This part of the dendrogram reproduces this family, almost perfect. And so on so far. Let's say the color code, it's the purity of a given cluster in architectures coming from these families. Okay. So just having shown that clustering can be employed for many things in our field, I just want to thank you. And I will be happy to reply any question.
Okay, thank you, Alex. So now, and thank you also for being in time. So now we have time for questions. Okay, so there are questions on the chat. I will read them. So one question is, can be hierarchical clustering associated with decision tree in machine learning?
No, I would say it's a different thing. I mean, decision trees are based on single features, let's say.
while in which for each feature you take a decision. Okay, let's say if a feature is bigger than a threshold, you are in a class. If it's lower than the other threshold, you are in another class. While here, the hierarchical clustering, it's dealing with all the features at the same time. It's taking us in the distances. So it's a bit different.
So there was Uriel raising the hand, but I cannot see anymore. So let me see on the chat. So Isabel Louise wanted to ask a question. So yes, go ahead.
Thank you for the interesting talk. I was wondering about the feature selection. For example, if we want to check the peptide conformation from a trajectory, is it useful to use many features like RMSD, hydrogen bonding, and also dihedral angles, or is it better to stick to one?
Well, what we do usually is to use many of them and then compare the results of the procedure.
Okay.
Because if your features, I mean, if according with our feeling, it's that these features should give you comparable results. Otherwise, you are having some problems.
Yeah.
So this is a kind of sanity check for our methods. We check in many different metrics and we see if they are giving you the same result.
Okay, yeah, makes sense. Thanks.
Okay, then we have a question from Daniel, who is also raising his hand. I will read the question, then Daniel just stop me if I don't read it correctly. Why the center points happens to be the outliers? I think it's referred to density peak clustering.
Okay, the idea is that the key point is the delta definition, in which you find the minimal distance to a point with high gas density. Okay, so the question is that the max, even points with high density have low delta if they are near another maxima, right? Yeah. So at the end, you are finding maxima in the density distribution. I don't know if it's clear.
Yeah, thank you, Alex.
Then a question from Andrea. Can you explain how to perform a validation for clustering?
Oof, yes, but it's a bit long. The question is that I decide not to include it because, well, mostly they are, okay, let me make a short introduction to that. There are two kinds of validations that you can perform. There are internal validations that depend on the structure of your clustering and external validations. In external validations, you are comparing with a ground truth. And you have metrics like normalized mutual information, for instance, for comparing with ground truth. In internal validations, you check some characteristics of your clusters, but the problem with these internal validations is that till the moment they heavily depend on what do you think your clusters should be, which it's not always the case. So the problem is that many cases, when you compare internal validations in cases where you have a ground truth, it doesn't happen that the best clustering is the one with the better internal validation index. This is a bit delicate.
Okay, then there are two questions from Suman. The first is how to find or choose an optical metric to classify a given set of data, because the efficiency of clustering the data by these metrics can depend on various factors. And the second question is how to pick an optimal machine learning technique like support vector machine or macro state model to explore a particular problem. Should it depend on the complexity of the system?
Okay. Well, for the first question, I would tell you, my experience is that the best feature selection is done by humans. Let's say human experts are the better ones in choosing features. And then once you have these features, you adapt your metric to these features. However, there are many, let's say, many techniques that allows you to automatically choose some features. All of them are relayed somehow in a kind of ground truth. So what you do usually is try to reproduce a ground truth that not necessarily it's in your system, but maybe in a similar system with some features, and then you just pick the features that better reproduce this ground truth. Once you have that, you can assume that in your system will happen the same and take the same features. Then, of course, the distance will depend on these features, mostly.
Okay, then
and the second question
sorry.
Sorry, I don't remember the second question.
The second question was how to pick an optimal machine learning technique to explore a particular problem. It's a very general question, but
that's a really, really general question. I mean, I also, because there is not one method, I would suggest you to really change methods in the same system. I would start with an unsupervised machine learning, and then if you have a ground truth, also try to use some supervised machine learning techniques. Then it really depends on your data. I mean, and really depends on how big is your data and how complex are them. In general, for quite big data sets, I would go for super neural networks, for instance. But in other data sets, it may not be the case. It's really the case dependent.
Okay, and then there is another question, which is from Zrabani, what kind of clustering would be useful to predict if we are on an unstable manifold of the phase space?
An unstable manifold of the phase space. Okay, a density-based clustering would give you an estimation of the free energy, in which with this free energy, you can check if there is, I mean, as far as you have sampling also the most stable states, you can check it. If not, there is no, I mean, you need that your data have some kind of trace that there is another state more stable. Otherwise, you cannot learn it from your data.
Okay, good. So I don't see any more questions on the chat. So I think we can continue. We can thank Alex again, and then we can continue with the next talk. If Alex, you unshare your screen, great. And then Ali should be able to share his screen.
Let me do that.
Okay.
All right. Can you see my screen?
So, yeah,
okay,
yeah, we can see it. We cannot see your face.
Hold on. Well, okay, it's not working now.
Okay, so I mean, we can hear you clearly. So
yeah.
maybe
Okay.
Fine,
okay.
as you prefer.
Yeah.
Okay, good. So the next talk will be given by Ali Hassanali, who is also from the ICTP. So please, Ali.
Okay. Let me. All right. Thank you very much, Giovanni, and I apologize for my video not working. Let me thank the other co-organizers in this activity, especially Angelo, for putting this together. And I also want to apologize for perhaps some technical hurdles that we've had today. We're all in a learning curve with respect to virtual activities. So what I want to tell you about today is regarding a problem that I've been interested in for many years, and that is the role of water. And the goal of my talk today is to try and convince you that water, the solvent around biomolecules in particular, plays a very important role in many things. And so just to get you warmed up and to whet your appetite, no pun intended, By the way, if you have any questions at any point in the talk, please feel free to interrupt. I'm happy to not finish my slides today. So, aqueous solutions are present in all our daily lives. I live in Italy, and so in Italy, I'm inspired by things like pasta and cappuccino, and there's some very basic fundamental processes in physical chemistry involving boiling your pasta or making a good cup of coffee that involve things like, the thermodynamics of surfactants, how they interact with soap bubbles, and how water interfaces with these processes. So just to build your intuition a bit and to calibrate you on a bit of some numbers, we can ask ourselves the following question, which is, how many water molecules, for example, are there in a cell? And if you take the E. coli, which is shown here in this bacteria, which is shown here in this picture, you can sort of count the number of different types of molecules that you would find there. So roughly you would have on the order of 10 to the 6 proteins. You would have roughly on the order of 10 to the 7 ions. And all of that is surrounded by a big bucket of water. And so the number of water molecules in an object like an E. coli is on the order of 10 to the 10. And so the one thing I would like you to take away from this talk is that in all these models and simulations that we show and we discuss, and when you think of processes in biology, you always have to remember that, of course, these objects are very well hydrated. And the water plays an intimate role in coupling to tuning the thermodynamics and the dynamics of the biomolecule. So let's get into something a bit more specific and ask ourselves, well, what's inside a brain? Okay, so these are images of, on the left side, of a brain of a normal person on the left, and on the right here is the brain of someone who suffers from something called Alzheimer's. So this is a neurodegenerative disease which leads to brain atrophy. And neurodegenerative diseases have been, mapped at the molecular scale to the formation of things known as amyloid fibrils. This is depicted here in this spaghetti-like chain of mesh of proteins. And if you dig even deeper and look even closer, what you will find is that these proteins, or these amyloid fibrils, are made up of things known as beta sheets. So these are, it's a secondary structure of a protein, which is held together by hydrogen bonds. Now, all this stuff, all these beta sheets, all these amyloid proteins are surrounded by water molecules. And so one of the questions that I've been interested in trying to understand is how water plays a role in stabilizing objects like this amyloid beta sheet. So it's actually really nice to maybe go back a bit in history and to see what people have been talking about this and thinking about. And if you go back, 3 decades ago to one of the first papers by Michael Levitt, who you heard today got the Nobel Prize in Chemistry several years ago, they performed, their group performed the first simulations of a protein in water and compared it to simulations that had been done in the gas phase. And it's very interesting to see here how the time scales of how MD simulations have evolved from 3 decades ago to now. And so on those 3 decades ago, doing tens of picoseconds was an enormous achievement, and it was. And one of the messages that came out from this paper, which was published in Scientific American, was that water plays a very important role, even though it makes it much more complicated to simulate a protein in tuning the structure of the protein. And there's been a lot of work over the last 3 decades from both the simulation side, theory, and also experiments in trying to understand how water plays a role. But there's still a lot of interesting open questions in the field. So this sort of leads to motivating a bit of what I'm interested in. And so I'm interested in creating aqueous solutions on the computer. And so I like to take different types of molecules. So things like the amyloid protein that I showed you earlier on, ions like the proton, I'll tell you a bit about this, and hydrophobic molecules like methane, and inserting them into water and trying to understand how these molecules respond to the feel of the water and vice versa. So how does water respond to the presence of these molecules? And secondly, does water just play the role as being a spectator in all these processes? So this probably doesn't need much introduction to you. You already saw a good introduction from Alessandro Laio this morning. So I'm interested in trying to design computational experiments where we try to understand how the complexity of water affects different processes. And so we use different types of flavors of molecular dynamics, some classical, when we don't have to deal with quantum mechanics of, for example, things like bond breaking, bond formation. And the real goal, and this is what I want to try and show you today, is to try to infer or come up with smarter water-based collective variables reaction coordinates, okay? And so in order to motivate this a bit, I want to show you, take you back several years ago. It's a movie that never gets old for me. And this is a very interesting problem in chemistry, which forms, the basis of pH. So there's this dynamic equilibrium between neutral water and its ionized products, the proton and hydroxide ion. And this dynamic equilibrium determines the pH of water. And so these were ab initio MD simulations that we ran many years ago. And I'm just playing the movie now where you're going to see some interesting things happen in the movie. You have the excess proton here on the left, which is the hydronium ion, so it's positively charged. On the right side here, you have the hydroxide, OH minus, which is negatively charged. And this is surrounded by a bucket of water. And there are some fluctuations going on here where you see, the proton here is exchanging with its partner in a strong hydrogen bond. And something interesting is going to happen here at the end of the movie, where there's some fluctuation in the liquid, which leads to these 3 protons jumping over at the same time and neutralizing the 2 ions. And well, the way we studied this problem many years ago is the typical thing that chemists do, which is you take out the VMD, you load the movie, and you try to look with your eyes and try to identify where the fluctuations are happening. What are the relevant coordinates? And I'm still a big fan of that. But what I want to try and convince you today is that there are a whole range of length scales and time scales of fluctuations in liquids like water, which are very difficult to see just by using your eyes. And so the combination of using, let's say, chemistry-based approaches with the type of approaches that Alex introduced in his previous talk, I think have a lot of prospects of understanding new physics and new chemistry. So the reaction that I just showed you is not necessarily biological per se, but there are many processes in biology where proton exchange plays a very important role. And this is an example of one where it's well known that the amide proton of diamide bonds of proteins, when it's exposed to solvent, can exchange its proton with solution. And this is thought to be catalyzed by the presence of hydroxide ions. And this becomes it's a very important experimental technique in order to identify which parts of the peptide backbone are exposed to solvent and which ones aren't. So again, for this type of a process where you have some large fluctuations involving your protein and your water, they become very challenging to just study by just using your eyes and typical chemical intuition based approaches. And so what I want to try and do today is tell you two stories if I get to it. The first one is related to how water acts as a lubricant for biopolymers, and some very recent efforts at using some more data science approaches to studying water networks. And if I get time, in the second part of my talk, I want to tell you a bit about this problem that I introduced you at the beginning, which is the optical properties of amyloid proteins, and some very interesting questions which you will hear about in one of the talks tomorrow on the importance of electronic excited states in biology. Okay, so let me get started and get to the problem. So, you've seen a lot of simulations today of different types of proteins. And the typical thing to do when you have a protein, let's say you have a protein in its folded state and a protein in its unfolded state, is to try to come up with, some order parameter that allows you to distinguish between the folded and the unfolded state. And one of the assumptions, of course, that one makes when you do this is essentially you're looking in some reduced dimensionality of protein coordinates, which I denote as P, and you're essentially integrating out the solvent degrees of freedom. And so I am interested in trying to understand how does that solvent modulate the free energy landscape of these polymers or proteins. And so there are lots of different ways in which one can try to interrogate or understand how solvent, affects the protein. As was mentioned in some of the talks, one of the first things you need to do is to try to come up with a relevant reaction coordinate, or an order parameter, or a collective variable that allows you to identify in your solvent degrees of in your solvent space some changes that are going on. And so I don't have time to get into the details, but one of the things that we've found to be extremely useful is, and that appears to give us much more information into the underlying free energy landscape of the solvent, is by looking at topological properties of the water network around the protein. And so the idea is extremely simple. It's been used previously in other contexts, that water is made up of a directed interactions because of the hydrogen bonds, and you can map this to nodes on a graph where you have outgoing and incoming edges, where the directionality of the edge corresponds to the direction of the hydrogen bond. And if you imagine a protein, and here this is a segment of this amyloid protein, you can think about lots of different water wires or paths that connect different donors to different acceptors. So every donor, for example, here the donor is an NH group and the acceptor is a CO group, can act as a source and a sink of hydrogen bonds, and therefore different types of water wires. So we've been studying the role of these water wires in changing the free energy landscape of proteins. And I just want to give you an example of how it can change the landscape. So this is some simulations of some MD simulations of a 6 amino acid segment of an amyloid protein. And the fringy landscape is shown as a function of the distance between the two termini on the x-axis, and on the y-axis is the radius of gyration. And what these different minima here on this fringy landscape correspond to are different states or conformations of the protein involving different amounts of hydrophobic contacts. For example, in this case, and in this case here in state A, and in state C, you have more hydrophobic contacts, whereas in state D and state B, the hydrophobic groups tend to be more exposed to solvent. Now the question is, well, where did the solvent go? How does the solvent affect the free energy landscape? So in order to answer that question, you need to know, or you need to figure out which degrees of freedom you should look along the solvent coordinate. And so again, just to give you an example of how the solvent can affect the free energy landscape, what I'm showing you here again is another reconstructed free energy landscape of the end-to-end distance between the two ends of this amyloid protein on the x-axis, and on the y-axis, this is a reaction coordinate that basically counts the number of paths that you can have between every donor and every other acceptor in the protein, normalized by a certain factor. And what you see here, I want you to focus on this oval region, is that there are several minima along this solvent coordinate that correspond to fluctuations in the solvent coordinate, but where the salt bridge is basically constant. And so in all these other, if I go to the previous slide, all these in the solvent coordinate is basically collapsed in this figure over here. But when you project it out, you see clearly that there are some other small minima that appear along the solvent degree of freedom. So now, in this system, you have 4 amino acids, actually 6 amino acids, and there are already over 50 different water wires that can form between every donor and every acceptor. I say this because just to give you a sense of where the problem of dealing with a lot of data comes into the picture. And so in order to dig deeper into this and to understand a bit better about how the solvent affects the free energy landscape of proteins, we had to break it down and go down to an even simpler system. And so this is a system that has been studied a lot in the literature, which is trialanine. And trialanine can be classified into structures that are, let's say, more alpha helical versus ones that are more beta sheet. And in this system, there are a total of 16 water wires. So there are 16 different paths that you can form between every donor of a hydrogen bond and every acceptor of a hydrogen bond. And so the question that we were interested in asking was, well, can I look at the water network around the protein, in this case it's a small peptide, and see, is there a difference in the secondary structure, alpha helix versus beta sheet? And so for this system, I guess it's not too much of a surprise. If you look at the distribution of the water wires that connect or that thread the alpha helix conformation versus the beta sheet, you see that, in fact, there's this gap between short paths and long paths for the alpha helix. This has to do with the specific constraints of the alpha helix. whereas in the beta sheet, because it's more open, you get this shoulder here in the beta sheet secondary structure that's not there in the alpha helix. Okay, so this is just looking at the water wire distribution around the secondary structure. But we can do a bit better, and so I started talking to Alessandro Laio, who has developed some, together with Alex Rodriguez, who you heard from in the previous lecture, who've developed some very interesting methods on trying to handle and cluster high dimensionality data. So one of the techniques that they've developed is this intrinsic dimensionality estimator based on the two nearest neighbors of every data point. And so I don't have time to get into this, but basically by using the two nearest neighbors of every data point, you can show that the cumulative distribution of this ratio can allow you to extract something known as the intrinsic dimensionality. What this physically corresponds to in the case of our water wires, or water network, is it tells us how many independent directions do the water wires fluctuate in. And so we applied this to this alpha trialanine system. So we have 16 wires. We extract the intrinsic dimensionality of the alpha helix versus the beta sheet, and you already see that there's a difference between the two. So this is already interesting, because the intrinsic dimensionality is telling you about in the water space what the difference between beta sheet and alpha helix is, and this sort of makes sense, right? Because beta sheets are more open, whereas alpha helices are more coiled, and so you might expect something like that. And then you can also construct free energy landscapes, dendrograms like the ones Alex showed you. And what you see is that if you look at the alpha helix versus the beta sheet, this is along the solvent coordinate only. You see that the alpha helix free energy landscape along the solvent is much more rough. It has a lot of ripples that are very faint, but it's markedly different from the free energy landscape of the water around the beta sheet, which is essentially flat. Okay, so there's only one minima. And so what this is telling us is that basically the solvent, at least at room temperature, in a very faint way, introduces some roughness into the free energy landscape of alpha helices. One more thing we did was to compare how our beloved water wires compare with standard ways of looking at solvent around biomolecules, so things like coordination numbers or essentially densities or GeoVars. And what you see here is that the coordination number is rather dull and boring. It doesn't really tell you about the underlying complexity of the free energy landscape. And this again sort of makes sense, because the thing that's contributing to creating this roughness of the free energy landscape is the orientational correlations of water dipoles around the protein. And this is something that's averaged out in the coordination number, but it's something that you get when you use the water wires. And so you can, now go in and this is why I think this combination of chemistry-based approaches and data science is extremely interesting. You can go in now and try to understand what these different clusters correspond to. And what you learn is much more nuanced. So what you learn is that the origins of the roughness of the free energy landscape is not made by very strict rules of this is purely hydrophobic, this is purely polar contacts, this is purely water. What you see here is that there are very, very subtle differences in the water wire distributions, the number of backbone contacts, and the number of side chain contacts, which in this case correspond to the number of essentially the hydrophobic interactions. And so all these various things contribute to the coupled free energy landscape of the protein and its surrounding solvent. Okay, so Giovanni, how am I doing on time?
Yes, you have, I think, 5 minutes. Or
5 minutes. Okay, fantastic. So in the 5 minutes, I will just tell you something very brief, but something that is, I find very interesting, and that is, so I've just mentioned to you a couple of indicators of what may cause the origin of roughness along the solvent coordinate. There are also other factors that contribute to the roughness, and something else that does is understanding how empty regions or empty space looks like in liquids. And so if you think of a thought experiment where you put a hydrophobic molecule or an ion in water, what you do is, if you're interested in the thermodynamics of that, well, you will sit yourself in a box of water, and what you will do is you'll wait for some time before you can form a cavity. And this cavity is an excluded region of volume. It's a hard sphere, essentially, where there are no water molecules inside here. And by studying the statistics of the formation of these empty spaces, you can learn something about the solvation thermodynamics of inserting hydrophobic molecules in water. And so this is something that has been studied to death by many people in the field. But typically, people have usually mostly only focused on, nice spherical objects. And we all know that biomolecules are not spherical cows. And so for a while now, I've been working on and thinking about how realistic regions of empty space look like in liquids. And I won't get into the details, but so this is just a sample obtained from a molecular dynamics simulation of how realistic regions of empty space look like in water. And so these are distributions here. You don't need to worry about the Met, just focus on the water ones, where these distributions tell you the probability of finding a normalized volume of void. It's normalized by the volume of a water molecule. And what you see here is that there's a peak at small values. So sorry, I should point to the water. And there's a tail to larger values, which correspond to these types of voids that look like this. So these are very branched dendritic shaped voids. And if you stare very carefully, you might be inspired to look for interesting resemblances of this to a basketball player, and this guy here on the left is a small dinosaur. And if you're even, you look even harder, what's curious about this is that these dendritic shaped voids look like small polymers, small molecules, chemical molecules. And so this is something that was done in collaboration with former postdoc, Narges Ansari, who's in the audience. And one of the things we asked was, well, can we find a shape of empty space in the liquid, so without a polymer, that resembles the shape of that you find around a polymer? And in fact, you can, and you can translate this into something more thermodynamics, which is the following, which is what is the solvation energy of solvating a hydrophobic polymer like this one here. And at the same time, you can also ask, what is the free energy required to create an empty space like this in water? And what's interesting is that you find that these two numbers are essentially consistent with each other. And so there are these rare fluctuations in water itself that create shapes that are consistent with the thermodynamics of solvating small hydrophobic solutes. Okay, so I actually don't have time to go through all this, so I'm going to skip it and would rather take questions. Oops. So, afraid my computer is frozen. Okay. Okay, let's go to the end. So I basically, I didn't get time to tell you about the first one, but what I tried to tell you about today is how essentially water plays an extremely important role in coupling to the fluctuations in biological matter. It affects the thermodynamics and the dynamics. And there's still a lot of interesting open questions in the field. And I think there's a lot of interesting synergies between combining chemical intuition-based approaches as well as data science approaches to learn new chemistry, new physics. So with that, thank you for your attention.
Thanks, Ali. Thanks a lot for being in time. So now we have time for questions. So just write on the chat if you want to ask a question. You can either write the question and I will read it, or you can ask me to speak. Okay, so there's a question. So from Davide, so what could be the outcome of the fact that little polymers can occupy the void left by water?
Okay, what could be the outcome? Well, so there are two things. One is there's a question of just thermodynamics, right? So you could it, yeah. So let me answer the first part. So you know, the way typically in the physical chemistry community, the way people typically have thought of solvation of hydrophobic molecules is that you need to there needs to be a huge reorganization of the solvent of water in order to facilitate the presence of the hydrophobic molecule. And what we're saying is that it's actually not so much like that for small molecules. It's that you know, the intrinsic fluctuations in the liquid can create shapes that can facilitate the presence of those molecules without having without paying a significant energetic cost. Okay, so that's the first, let's say, implication. Yeah.
There's another question from Davide, which is maybe ready to read it. Could it have an outcome in the drug discovery field?
Could it have an outcome in the drugs? This is a well, a difficult question. Well, maybe. I think the question is in the drug discovery field. What would be interesting to think about and wonder is when you have a drug interacting with a protein. How are the dendritic voids and empty spaces around those two objects talking to each other? Okay, I think this is not understood. You know, there's been quite a bit of work trying to understand how dielectric properties, for example, between two proteins or a protein and a drug change when they come into close proximity. But where I think this has a possible relevance is, you know, really trying to understand how these fluctuations around the drug or around the protein may couple to each other.
Okay, then there's a question from Lorenzo that is, with the aim of reducing the computational costs, what do you think about hybrid solvation? I mean, for example, a shell of all atom water around the solute, all surrounded by coarse-grained water molecules.
So I think it really depends on what your questions are and what you're interested in. And so I'm generally not a fan of coarse-grained models. I understand the need to do them. You know, what I would say is the following. I think let's say the building evidence from experiments and theory is that water around biomolecules can feel orientational correlations up to about 15 angstroms. Okay, so if you think that these types of long-range orientational correlations will affect the, let's say, the process that one is interested in, then I would say one has to be very careful about how you choose the thickness of the hydration shell. Okay, but as I said, I don't think they're, I can equally buy the argument of, if you want to simulate large complex systems, it obviously makes sense to do some type of coarse graining. So I think it really depends on your question.
Okay, then there's a question from Andrea. In atomistic simulations, how different water models, for instance, TIP3P or SPC, affect these properties and or analysis?
Yeah, that's a fantastic question. And so basically, the bad news is that TIP3P, which is the water model that's used in many biosimulations, is quite horrible for water alone. Okay, and now it depends. Again, is sort of similar to my previous answer. It depends a lot on whether one is interested in thermodynamics or dynamics. For thermodynamics, I think choosing the water force field with the appropriate protein force field, you sort of more or less are able to converge similar results when you look at the properties of the protein. Now, on the other hand, if you look at if you're interested in understanding the properties of the solvent around the protein, then it can be very sensitive. Okay, and it can vary from one water model to another. So, yes, results can be sensitive to the choice of your water model, in particular the dynamics. For the things that we looked at, which are these topological properties, they are much less sensitive because it's, most water models have more or less gotten, they're mostly tetrahedral networks, okay, with some defects and so forth. So that's more or less correct.
Okay, then Sudarshan would like to know, what tool did you use to get the hydrogen networks around alpha helix and beta sheet?
So I once had a very good PhD student who left for North Korea. And he wrote all sorts of things for me in C++. But there are lots of tools available, ours and much better, much more efficient ones by other people who are developers. So, yeah.
Okay, then there is another question, but it's a duplicate. Basically, what is the basis for the selection of water potential models?
Yeah,
so,
yeah, I think this is it's a repeat. So
yeah.
Okay,
good. So any other questions from the audience or comments? Okay, there is another question from Marcelo. So is there a linkage between the void of water molecules and the polarization of them? If so, how would it possibly be connected with the molecules you've shown?
Yeah, that is a great question, and so in fact they are. We have some unpublished or ongoing work right now that examines the extent of charge transfer between water molecules close to a void versus being far away. So you can think of a void as a sort of an enclosed short length scale region of analogous to an interface. Okay, and essentially there is a polarization at these interfaces and around these voids. And where I think it will play a much bigger role is when you start to introduce charged molecules. So if you have a molecule with a, well, it doesn't actually have to be charged. Something with a dipole moment, a quadrupole moment, may couple in a non-trivial way to the void and its corresponding charge polarization. But that's a good question, yes.
Okay, good. Then there's another question from Jamal that is, what kind of interaction will it have in empty spaces?
Sorry, which kind of interaction?
Will it have in empty space? I don't know if Jamal wants to clarify.
Can you, can you clarify what you mean?
Yeah. Okay, I cannot hear Jamal, so. Okay, good. So maybe we can thank Ali again and then proceed with the next talk. So the next talk will be given by Angelo. So Angelo, I think you can share your screen.
Yes, I unmute. Okay, share screen. Yes. Can you see it? I think so.
Yeah, clearly. Okay. So the last talk of today will be given by Angelo Rosa, who is also from the PhD in Physics and Chemistry Biological Systems at CISSA. So please, Angelo.
Yeah, thank you, Giovanni. Thank you all for listening, especially for asking very nice questions. So I'm very happy about that. As I said, it's a sort of experiment for me, this event. So it was very nice what Ali said at the very end, because he said, I don't like very much coarse grade models. And actually, instead I do. And so that's a nice way of sort of connecting to the previous talk, and also to connect it to the talk which Christian Micheletti has done. In fact, the first part of my talk, it was very short, but it will illustrate a little bit sort of a few things about polymer physics in general, and why actually you can use coarse grain model to learn a lot about the physics of polymers, okay? And why they make sense. I mean, this is probably trivial for many of you, but since this at the origin was meant to be a school, so I preferred to keep also some pedagogical opportunity to say something. So briefly, I mean, I'm sure here everyone knows what a polymer is. So basically, it's a big macromolecule. It's a big molecule, and is made of out of a polymerization process. So you just can put one after the other some minimal units, which conventionally we can call just monomers. So they exist in different, I think they exist in different architecture, and also what is very important, in different states. So if polymers are quite dilute in your solution, so you have a sort, basically you have a gas, the properties are very similar to the ones of a gas. Then if you start increasing concentration, you go to a liquid-like state. And then if you increase the concentration even more, you got something which is pretty much like a solid state. For instance, rubber basically is made of a polymer, so joined together, and but it looks like a solid, at least on some time scales and length scales. Of course, now we have biopolymers, and we have seen a lot this morning, also this afternoon. So biopolymers are proteins, nucleic acids, and actually nucleic acids, it will be part of my talk in the second part. And we have chromatin. So chromatin, actually more chromatin than DNA, will be the second main actor of the actor of my second part of this talk. And the chromatin is basically is DNA plus some proteins. So, but I mean, to all extent is a polymer itself. So now, why? I mean, what is polymer physics? So polymer physics basically is the study of polymer conformations. So if you take, for instance, a very simple polymer, which is polyethylene, the single monomers, I mean, the two single monomers when joined together, they can exist into actually in 3 different states, 2 gauche, according to the orientation of the bond, and 1 trans, which has a sort of smaller energy. And depending on how these bonds are arranged, I mean, a long polymer chains can attain a high number of conformations. So it's an ensemble of conformation. You don't have only one unique conformation. And so the polymer physics, I mean, the object of polymer physics is the study, the sort of the statistical mechanics of starting from this conformation. As you can imagine, based on the N is the degree of polymerization of the polymer chain. So these polymer conformations grows rapidly, basically they increase exponentially fast. And so then you can use the tools of statistical physics to study any polymer problem, I mean, where physics enter as an actor. So there are the basics idea were firstly formulated in the, I mean, between the 30s and the 60s of the 20th century by many outstanding people like Kuhn, Huygens, Schockmeyer, James, etc. While let's say more sophisticated stuff like the study of polymer solution, polymer melts, and the role of entanglements. We have listened to the previous talk by Christian where he talked a lot about entanglement. Were formulated by many people like Edwards, Descloiseau, and especially De Gennes, who got actually the Nobel Prize for his work on such kind of stuff. Let's say, if you want to formulate a polymer model a physical model for polymers, you start from something very simple. And there are basically two simple polymer models. One is the freely joined-in chain, where basically the joint between two monomers is completely free to rotate into the space. And another model is the freely rotating chain. So now this angle theta between one bond and the next one is fixed in space, is not allowed to take any value between 0 and pi, while you have still some freedom of orientation in the azimuthal angle phi, and which can still move between 0 and 2 pi. And now what you can ask is, what is the typical size of this polymer? And you can do the calculation exactly in this case. And you can see that in the first case, basically, is just if you want the free joined-in chain is just like a random walk in space. So just it increases linearly with number of monomers by some prefactors, which is the average bond length between 2 monomers. While in this other case, the chain is slightly, in general, depends on the value of this angle theta. It's more or less stiff, but still it increases like n in terms of the number of monomers. So now all these 2 models actually, let's say they belong to the same universality class, in the sense that you can define a typical length scale for each of the 2 models, which is called the Kuhn length, which is a measure of how, let's say, of the rigidity of the polymer, of the local rigidity of the polymer. And then everything, the typical size of the polymer can be recast in this universal form. So, and where nk is the total number of Kuhn lengths, of Kuhn segments, if you want, of your polymer. So and this tells us a very important thing, that somehow these laws are sort of universal. You can still have basically the same model describe the polymer chains regardless of the microscopic details. And this is, yes. And this tells us a lot, about what polymers are. Basically, polymers are fractal objects. And if they are fractal objects, of course, this is very nice, because now we can employ to study them all the ideas that comes out of fractality and the self-similarity. And basically, we can employ things like those in order to study models now for real chains. Now, by real chains, I mean the following. In the previous slides here, I have basically, any 2 monomers can just occupy the same portion of space, right? there are no correlations, so two monomers can overlap together. This is of course not very realistic. While we need to exclude those conformations which where any two monomers can share the same portion of space. And in this case, so it comes, I mean, something which becomes rapidly more difficult, because now all these kind of models cannot be solved exactly. And so in order to get an idea of what's going on, we have to employ either mean field theories like the ones that were formulated by Paul Flory, another Nobel Prize winner actually for his work on the physics of polymer, on statistical mechanics of polymers. So you do, so if you apply his tool, you know, I mean, you can get that the typical polymer size now is no longer increasing like the square root of the number of monomers, but it's a different power law, meaning that the polymer is more swollen. Or you can employ, as I said, since polymers are fractal objects, you can employ something very sophisticated, like basically renormalization group. And sorry, I just want to, okay, like a renormalization group. And if you employ renormalization group, it means that you can basically use all the tools that were derived in the 60s by guys like, again, the Jean basically, and the Cloiseau. And in order to, what you do basically, you can have your polymer chain, then you construct sort of super monomers in such a way that you make a coarse graining of your polymer chains. So what you do, you coarse grain your monomer, and you also coarse grain the interaction. And if you do that, you can apply iteratively this procedure in such a way that you arrive to an estimation of how the chain grows inside with the number of monomers. And the result actually becomes pretty accurate, because you get an exponent which basically confirms the mean field, but it corrects it, because now this is rigorous. And at the same time, it tells you that, an important thing, that basically you don't need too much information on the, let's say, on the local interaction, but what you need is some sort of simple model, because then all interactions are renormalized, so then you get the right answer. So that's why somehow you can translate all these ideas also in computation. You can use, especially for the large scale behavior, you can use a coarse grain model, and you can study big system. One typical, and then I go rapidly to an application of this idea where I can employ actually, where I need actually to employ a coarse grain system, because the scales are too big and the system will be too large, that a fine grain or a full atomistic simulation is impossible, or full atomistic approach is impossible to do. So the system, yeah, sorry. Okay, sorry, the system that I have in mind is the problem of chromosome folding. So what is the problem of chromosome folding? So so far, Christian also has mentioned here in this morning, so you have DNA, but in cells, I mean, DNA is quite complicated in the sense that you have not only bare DNA, but you have DNA plus some proteins. These proteins are called histones, and the histones, sorry, the DNA is wrapped around the histones, and they form a sort of necklace. This necklace is called the 10 nanometer fiber, because basically let's say the average diameter of this fiber, DNA plus protein, is about 10 nanometer. Then what it is observed that in some specific condition, this 10 nanometer fiber is folded into a super fiber, let's say, which is called the 30 nanometer fiber, because again, the diameter of this fiber is around 30 nanometer. And then, I mean, under some condition, these things take the form of chromosomes, some of these, let's say, these X-shaped chromosomes, X-shape that you can find in textbooks. Okay, so that's basically the biology of chromosomes. Now, what is interesting about that is the following. It was observed now more than actually at the ending of the 19th century by these two guys, Carl Raab, two German biologists, Carl Raab and Theodor Boveri, that actually chromosomes are not sort of randomly organized inside the nuclei of the cell, but they take a very specific shape. So that at the time was really generic in the sense that they were studying that from by using optical microscopy. So the study, nonetheless, I mean, they could predict something, they could observe some things, but I mean, it was not very quantitative. In order to get some, I mean, a more quantitative picture, we need to go to the 70s, I think, where these two two German guys, actually two German brothers, they invented a technique which was very clever, in my opinion. So what they did, they used some laser beam on the nuclei of some cells. Don't remember which cells, but were eukaryotes, so cells with a nucleus, and they beamed the nucleus of the nuclei of these cells. So they said that the following. So if the hypothesis of let's say of Rabol and Boveri were correct, so namely if chromosomes are not just randomly organized, but they have a structure inside the nucleus, so then we should observe the following. So if you imagine that the chromosome are just many chromosomes, just like a tangle of structure of DNA of fibers, and if you beam them, so then sometimes you destroy, I mean, the laser is so strong that destroys DNA, so it makes a breaks. So, I mean, if you do it just in some random spot, then sometimes you hit a chromosome, sometimes you hit another chromosome, because they are all mixed like a spaghetti dish, okay? Instead, if they are organized, then you should, I mean, and if you beam them again, then you should only destroy a few of those, just a very small number. And actually, that was the outcome of the experiment, okay? That was done back in the 70s. So this picture of non-random organization of chromosome was confirmed actually by another technique, more sophisticated actually, which is called fluorescence in situ hybridization, where basically you can design specific probes for selected sequences, for selected DNA sequences on chromosomes. You can you can design probes with different colors according to the sequence you want to probe. And then at the end, by using high fluorescence microscopy, you can basically see chromosome inside the nuclei of the cells. And so this is the picture you have here on the right bottom corner is a sort of reelaboration of one picture that they observed by high fluorescence microscopy through fish. And here each color corresponds to different chromosomes, meaning that the chromosomes actually, they are not like spread inside the nucleus, but they are organized in some specific portion of the nucleus that these guys call chromosome territory. And actually the presence of territory is quite important because it correlates with, so it's not random inside the cells in the sense that they found some correlation between big chromosome and small chromosomes. Sometimes big chromosomes, so I mean, most of the cases, big chromosomes are found towards the periphery of the nucleus and small chromosomes toward the interior, and they are functional. So in the sense that if you destroy this kind of organization, or if you damage this kind of organization, this is associated to potential diseases and also to the death of the cell. So and this is, for instance, another observation they made, that actually mammals, which are, let's say, which are very similar or very, I mean, close from the evolutionary point of view, but they have developed for reasons different habits. Like, for instance, one mammal is diurnal, one mammal is nocturnal. The chromosome organization in those cells, for instance, the retina cells, is different, and it responds to these to how these mammals have evolved in the course of their life. And this is another technique that was developed. This is actually much more recent than fish, and this is called chromosome conformation capture technique. So these techniques allow you to detect how frequent the DNA interacts in the nuclei of the cells, and this is done in this way. So you use formaldehyde in order to make cross-links of DNA inside the cells. Some enzymes, which are called restriction enzymes, that cut all these fragments. And then you do a bit of, let's say, post-processing of these fragments. Basically, you amplify these fragments, and you detect the frequency of how much frequently these fragments appear. Okay, and the idea is that fragments which more frequently interact, they should appear more frequently in your statistics. Now, of course, you have a lot of these contacts between fragments which are very close in sequence. Basically, the fragments of DNA which are very close in the sequence of chromosome interact frequently, and this appears in this contact matrix, so where the diagonals is very populated. But there are also non-trivial contacts, in the sense that contacts which are so there exist contact between DNA fragments which are very far in sequence, but they are close in space. Okay, and this concept tells us that there exists a non-random organization for chromosome. So and this tells us, if you want, this complements what what Fish has told us, that there is no random organization of chromosomes. So now, can we understand that? And that was basically the purpose of my investigation of what I will present now. Can we understand, by some just simple physical mechanisms, the presence of non-random organization of chromosomes inside the nuclei of the cell? So now the problem is interesting from by many point of views, and it's because of the following. So if we take just an ordinary human cell, so the ordinary human cell contains about 6 billion base pair of DNA. So it's about 2 meters of DNA inside each cell. And so this cell is contained inside the nucleus, which is only 10 microns of diameter. So you have, let's say, you have if you want to make the proportion, it's like stuffing, a 100 kilometers rope inside an ordinary backpack. So, and as also Christian has pointed out before, so there is a lot of problems with that, because you create a lot of entanglements. So that's why the problem is so interesting from the physical point of view. So if you want, this is another proportion. It's the same distance as between the center of the sun and the Uranus, and the nucleus is just the Earth. So there is a really huge gap in length scales. So in order to better understand the problem from, to cast the problem into a physical problem, we just go back to what happens during the cell cycle. So basically the chromosomes are very dynamic. So you have 2 phases during the life of the cell. One phase is called mitosis, and it's where chromosomes take this peculiar shape, like this X shape. And then you have interphase, and it's during interphase that chromosomes behave, as I said, territorial. So they form this territorial organization. And what it happens in between during mitosis and the chromosomes when they enter interphase is that they start to open from this X shape they have during mitosis, and do some dynamics inside the nucleus, and then form territories. So if you want to get a clue, what's going on, then you can do that. Actually, it's what we did at that time with Ralph Everers, who is also going to give a talk in this conference, this event. Was the following. So basically, we simulated let's say the transition from the mitosis to interphase, in order to see if we saw, by using a polymer physics model, in order to see if we could get something about the formation of the territories, an idea why territories form. In order to do that, we use now, as I said, a very coarse-grained model, because, as I said, each chromosomes, this is for the typical mammalian cell, and each chromosome here, it's about 100 million base pairs. It's an enormous system. No way to do that, let's say, by using the standard simulation we saw before, like using water, explicit sum, and things like that. So we need to do something very coarse-grained. And the level of coarse-grain we used was at the level of the 30 nanometer fiber. Namely, if you do some mapping, means that we used a chain of bits for model one single chromosome, and each bit mapped to 3,000 base pairs. So if you do then the proportion, that means that each chromosome here, each chain is about 30,000 monomers. At the time, we used 4 of these objects, which are totally, they are all the same. It's an homopolymer, so there is no chain, there is no information from the sequence. It's very basic. So in the end, the whole simulation contained about 120,000 monomer particles. And the chains were arranged, let's say, randomly inside the box with simulation, with periodic boundary conditions. And otherwise, we arranged the chains as, by using a very simplified model for the mitotic organization, and let the simulation go. What we used then was a standard Brownian dynamics simulation of a generic, as I said, a generic polymer model. It's called the Kremer-Grest polymer model. Basically, as I said, it's a Brownian dynamics simulation. So you have it's a Langevin equation with some potential between the monomers, with noise with zero average and standard and delta correlated in time and in the monomers. So the force will, let's say, it's very simple and it's something very standard using polymer physics. So you have chain connectivity. Of course, this is very important, where you use what is called the Fine potential, which is, if you're unfamiliar with that, it looks a bit strange, but basically this potential was done in such a way that two monomers, two bonded monomers cannot go too far. We don't use harmonic potential because the presence of this logarithm term, basically, if the distance becomes too far, then this potential diverges. So that's why we don't want that, because that's a crucial part of the story. Then we have some standard bending stiffness for the chromatin. We just tuned this parameter in such a way that we matched some experimental observation from the literature. We imposed the number of monomers and the size of the box, inside of the periodic box, in such a way to mimic the original. What's the average density of DNA or chromatin inside the cell. And then we imposed hardcore repulsion between monomers. So basically, when these two monomers fluctuate and they do bump into each other, they cannot cross. And that's why it's also important actually to use Fine, because if you use a potential which is for bonding interaction, which is just harmonic, basically, you can, basically what happens, you can break topology, because the monomers in principle can go quite far apart in principle if the potential is not too strong. And then you can have two chains can pass through each other. We don't want that, okay? So that was our minimal ingredient. So if you do that, basically you let the simulation run under these, starting from this initial conformation, and you end up with something which looks like that. So basically each chain maintains, from the visual point of view, maintains its identity. And as you said, it's very similar to what can you observe inside the cell. So you see territories. But can we be more quantitative than just pictures? Yes, one important thing here is how, which is I think is a bit sometimes underestimated, but it's a very important point. What is the typical time scales you can do in this simulation? Because this is, of course, coarse-grained simulation. So basically, you need to fix in some way, basically diffusion coefficient of your monomer. In order to do that, we did the following. So in the literature, we found some data on where people were monitoring the mean square displacement of a single gene on some I think it was yeast chromosomes in time. So they used microscopy in order to monitor how these things displace, and they measure the mean square displacement in time of this locus. So then what we did, we actually simulated the same system using our model, and we just fixed the time scale in order to match the experimental observation. Actually, here there is a point. What we did basically, which was only a shift in the X scale without shifting on the Y scale. So that means that we had the complete control of the spatial scale. There is no fit on spatial scale. There is only fixing the time scale. And so by this so if you want, I can give you more detail later. So by this protocol, we were able to show that with this setup, you can basically reach. I mean, at the time was like about 3 days in real time. So that was how we fixed the time scale of our simulation. And that was quite important, because then you see that you can reach macroscopic timescales, and so all these things make sense. And then we can compare to experimental data. So these two pictures are the following. Now I'm going to explain a bit more in detail. So here the lines are the results of our simulations. The symbol are the results of experiments. So in the top panel, experiments are the mean square spatial distances between two loci on some chromosomes. So sorry, I don't know if I can. Yeah, so the different colors here, sorry, it's not indicated in this figure, but the different colors means different species. Okay, so red colors is yeast, Saccharomyces cerevisiae. Here blue or cyan is men, Homo sapiens. The green one is also Homo sapiens, and the brown is Drosophila melanogaster. And the lines are the results of simulations for the same quantity, namely the mean square spatial distance between two loci on the same chromosome as a function of the genomics distance between them. This is, let's say, it's a standard quantity you can measure in polymers to basically to read the statistics of your polymer chains. And as you can see here, that the results of simulations, which are given by the lines, match the results of the experiment. So we were able to provide a quantitative. So basically, our model is able to summarize in a quantitative way the outcome of its experiments for the structure. Another, of course, I mean, one set of experiments is not enough. So we saw by the same simulations what it happens to when you measure how two loci, the same pairs of loci interact in space by comparing to results of this other technique, chromosome conformation capture, that I mentioned at the very beginning. And also here the result is in agreement with the experiments. So I guess Giovanni is appearing. So how much time do I have? 1 minute. 1 minute? Okay, no, I'm done. Yes, and what is important here?
2-3 minutes is okay.
No, it's okay. I can finish here. It was just a part of showing the results of this simulation. So what's important here is also, I mean, this kind of behavior you observe here, because it's not by chance. So we have an explanation, a quantitative explanation why I observe. I don't have the time to go into the details. I mean, you can contact me in case you are interested, or we can discuss later in the questions time. So the kind of exponents you observe here, which is let spatial distances increase like 2/3, and contact frequencies decays 1/L, tells you that basically this is a non-standard polymer systems. It's not Gaussian, it's not self-avoiding walk, it's something completely different. We have a quantitative explanation and a quantitative quantitative model basically from first principle why these things are like this. Well, if you want, I can tell you later on another day. But this something is very interesting, because then it allows us to do some specific mapping and to construct a more coarse-grained version of this system and to simulate bigger system. Otherwise, we can do the opposite, and we can do also some fine graining by starting from the same model but going to smaller scale. because now that we have sort of an intermediate scale from 30 nanometer to something larger, you can do also the opposite and then go below by using some very straightforward fine grain. And that was, we did it a few years ago. Sorry, I don't have time to go into details, but the fine grain basically allows you to study smaller scale in a very systematic way, and also to explain how certain genes behave in a very simple way. Basically, this is all a mechanistic effect. So at this point, so I just want to, this is the explanation of blah, 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 what I was mentioning before, but I just want to conclude by acknowledging, the two people that were really involved in this work. First of all, Ralph, which is going to talk in a few days, I think on Wednesday. And Anna Maria was a postdoc here in SISSA. He left a few years ago and now is working in Molecular Partners AG in Zurich. And of course, I want to acknowledge many computational resources I use in these years. So thanks for your attention, and thanks Giovanni for chairing the session.
Thanks, Angelo. So now we have time for questions. So as usual, just write your question on the chat.
So just a question. I don't know if you were able to see the arrow. No.
Yeah.
Ah, yes. Okay.
Yeah, I can see it now.
See. No, just because I didn't know.
So questions or comments from the students, it's very important. Curiosities. Maybe I will start with a question. So how expensive are these simulations?
Well, I don't know. No, I know, but instead, it can be very expensive actually. The first simulation I did, but it's now quite old. It's about, I think I spent about a month on some cluster for simulating these. You can still see the screen here, right? Yeah. So when I did the simulation, when I was a postdoc in Dresden, so that was pretty expensive. I think I spent about a month for doing these simulations. If you want to relax, first of all, if you want to relax completely this system, this is impossible. So then you can relax up to some length scale, and then and that was the point of why these things work actually, because it actually is not relaxed. That's why I didn't have the time to explain that into details. But then you can do, as I said, you can construct a protocol, which is in order to let's say to use a sort of multi-scale modeling, and that is much less expensive, and you get the same results, but then you get the complete, let's say, this confirmation from basically with a few days of computation. So by then you are using Monte Carlo. So you are building another model which maps into this, but it's not molecular dynamics in Monte Carlo, so you don't have access to dynamics. Of course, then from the same structure that you have built by Monte Carlo, then you can do the dynamics if you want. But then, by the end it takes a lot. I
see. Then, so there is a question on the chat from Fahad asking if you could explain the L to the 2/3 law.
Yes. So actually, as I said, so I was a bit, well, I was not really careful here, but it's a bit, so because I didn't have the time. So, but the fact is the following. So if you, so the idea is the fact is the following. So you start from these conformations, right? Which are obviously not in equilibrium. So then you let these things relax to some point, whatever this point is, and then you end up with this. But then again, as I said before, these conformations are not equilibrated in the sense that they are still keeping memory of the original conformation. So basically here, what you see here somehow is what you have put into the initial state. So then you say, okay, but this is a bit disappointing, right? Because, I mean, so what's that? And the fact is the following. So, and that explains also these exponents. So suppose you do the same, this kind of sort of experiment, let's say Gedanken experiment. So what you have here is you start from some conformation which is not in an equilibrium, is this kind of rods, if you want. Then you know that just by entropy, sooner or later, you should end up into this kind of conformation where everything is completely mixed, just because of entropy. This is something that is perfectly known, I mean, where polymer have to go. But before doing that, I mean, before reaching this state, you have to go into something which resembles more like this, where sort of you have some partial, let's say, relaxation of the structure, of the original structure, but you don't have the topological relaxation of the topology, namely, these things, these objects at the very beginning are not entangled, I mean, let's say, are not linked with each other. And so they maintain this linkness, let's say, for a long time, right? So if that's the case, then you can just say, okay, then you can estimate how much it takes to relax these topologies. It's very long time. But then I can say, okay, then I don't care about the fact that these chains have some ends. So I can just forget about the ends, and I imagine that my system is not a system of linear chain, unlinked and entangled linear chains, but are a system of ring polymers, where rings, I mean that I unlinked and unknotted, because basically, if you want, it's really the same thing. And so then I let these things to relax. Okay, if you do that, then I can say this system of unlinked and unknotted rings have something to do with the simulation I did before, let's say for the for non-equilibrated linear chains. And if you do these experiments, by exactly the same simulation, I mean, the same setup, the same model, you end up with something like this. So basically, equilibrated rings, okay, behave like let's say, non-equilibrated, unlinked linear chains. So this is the same measurement I showed you before. So the mean square internal distance is between two loci as a function of, if you want, of the genomic distance. So now if you do that, now we have a reference system which is at equilibrium, because but it's not linear chains, it's ring polymers. And then you can, now there is a lot of literature on that. So now a system like this has to be completely different from the equivalent system of linear chains. They should be more compact, and they should display precisely this exponent. Namely, they must be compact if you want. They are space filling, because this exponent tells you that ring polymers are space filling, and the contact frequencies between loci has to decay like 1 over L, and not like 1 over L to, for instance, 1.5, which is the typical behavior of a random walk, or what you would expect in between contacts on a linear chain if the chain was a random walk. A self-avoiding walk would be even worse, would be even more steep than that. So if you use a model for rings which are naturally, if you want, territorial, because they are naturally compact, so then you get an explanation for this kind of exponents, which is quantitative in this distant equilibrium system. Then you can do thermodynamics, so statistical mechanics of this. And now there is a lot of literature on that. Probably Ralph will mention a bit that. This I don't know. So I don't know if I answered the question.
There's another question by the same person. That is, do your simulations agree with loop extrusion theory?
Yes, the good question. There is no loop extrusion imposed here. And I think my answer would be no, in the sense that this is something that actually this pertains to a non-equilibrium mechanism. Which means that I don't expect that to become compatible with that. But of course, you can impose that. It's pretty straightforward, because now you have, if you want, is a sort of ground model, then you can impose loop extrusion. Then it's another question is if the structure you get by loop extruding, by using loop extrusion on some initial structure for some time, does it look like this one that I'm constructing here? Maybe this I don't know, but it's an interesting question. Actually, we are thinking about in this direction. But so that's another point. But I agree that is now loop extrusion is quite popular subject. So I agree it's an important point.
Okay, good. So I don't see any more questions in the chat. So if there are no more questions, I think we can thank Angelo and all the other speakers again. And then I saw Adi appearing. Maybe do you want to say something? No? Okay. Okay, good. So thanks to everyone. And yeah, the school will continue tomorrow with the advanced lecture. So see you tomorrow morning.
Thanks everyone for participating. Ciao, bye bye.